So I'll welcome everyone to start sitting down and getting back in their scientific hats on and everything, because we're so thrilled to have Abdel here coming in from Europe to give our second keynote. So Abdel's an assistant professor in the Department of Psychiatry at Amsterdam University Medical Center, and I think most everyone in the room knows his work really well, but just for those who don't know all of it, I'll say a few things. So I look at it, and our local group has just been so impressed and followed his work for many years now, because he's led and collaborated on so many, both genetic discovery, GWAS and related analysis, but also on the discovery side across so many outcomes that we're interested in, from health behaviors, mental health, but definitely social traits, educational attainment, and then also this other lane of research on understanding population structure, and in particular migration patterns, and how essentially the social becomes the genetic becomes the social. That's the title of his talk today. So he was a recent awardee of the Fuller and Scott Award for Early Career Contributions at BGA a couple of years ago. His work's been cited over 20,000 times, very high impact on many of our fields. So join me in welcoming Abdel.
Well, thank you, Jason, for that very kind introduction. Right. Let's see. Oh, yeah. So I like to look at larger patterns of variation in, especially in big genetic data sets and in combination with geography. The first article where I touched genome-wide SNP data, I was tasked with cleaning the data set. So I looked at large patterns of variation that allowed me to see like quality differences in the data set, identify those, and the next step was to look at what the larger patterns of variation mean, because these are things that you have to control for in a genome-wide association study. So I did a principal component analysis. That is an approach that summarizes a large number of measurements into the largest patterns of variation. And if you do that in genetic data and you don't have these big quality differences left, then these large patterns of variation reflect ancestry differences. And if it's a relatively homogeneous population, with, let's say, all of them have Dutch ancestry, and you plot those principal components on a map, you get these very nice geographic patterns. This principal component analysis does not know where these people live, but the closer you live to someone, the more likely you are to share more ancestors with that person. And so is this also a mouse? No, it's not. The largest patterns of variation is in that first map, the most left map, that has these three major rivers that go through the border between the red and the yellow. So this is a geographic barrier, but there's also a cultural barrier, namely the Catholics and the Protestants. And so all the way on the left, you see the map of the Catholics and Protestants about 150 years ago. And Catholics have been living quite segregated from the Northern Protestants for many centuries, and also having only Catholics having babies with other Catholics, Protestants only with other Protestants. There is a saying in Dutch, twee geloven op één kussen, daar slaapt de duivel tussen. Are there any Dutch-speaking people in the audience? No, not at all. So that means two religions on one pillow, the devil sleeps in between. So if you have this strong associative mating on religion for many centuries, then these genetic differences are maintained and diverge, and we see also that these north-south differences in the Netherlands, they are strongly in line with European north-south ancestry differences, with this principal component correlating with things like height and eye color. So I wrote a number of articles trying to put these ancestry differences and the relationship with geography in a historical context. This was done in about 7,000 Dutch individuals. And then more recently, it works again, yeah, thanks. So more recently I've been able to look at larger data sets, the UK Biobank in particular, which is 500,000 British individuals. And there I had the statistical power to look beyond ancestry at geographic patterns of genetic variants that influence complex traits. And when I controlled for ancestry, I saw that the strongest geographic patterns of genetic variation aligned with socioeconomic status, and that's what the rest of the talk is going to be about. So I have to underscore that all of the things that I will be talking about are based on studies that only have subjects of European ancestry. We filtered out people of non-European ancestry because of potential confounders, and so this says nothing about differences in socioeconomic status between different ethnic groups. That's a whole different story. So social inequalities, they have been decreasing between countries for a while, but within countries they have been increasing, especially since the 80s. There are social and economic forces that play a role here. Technological changes have an influence on this. Technological advancement allows for companies to consist of fewer people and make more money. And this paper, so this is the paper where that I was hinting at earlier, suggests that genetic effects are playing a role in this story as well. So we were not intending to write a paper about social inequality or social stratification. We were interested in the geographic patterns of a whole wide range of polygenic scores for a wide range of traits. But there was one trait that was the big outlier that showed the strongest regional differences, and that was the polygenic score for educational attainment. And the patterns that it showed, like if you plot it on a map, you see the same map as something called the Townsend Index, which is just a measure for how well a region is doing economically. And then on the right, you see the map for overall health. So mental and physical health outcomes show a similar map, but the polygenic scores of those same mental and physical health outcomes don't show this geographic pattern. So this implies that there are environmental influences that are causing these differences between regions in health outcomes and economic outcomes. So you have the genetics and environment clustering in a way that makes it difficult to tell them apart. So these show these health differences, the relationship between regional economic deprivation and life expectancy is a clear relationship, and then the two most deadly diseases, heart disease and cancer, it matters for the region how rich it is for these outcomes. Same goes for mental health. So you have these genetic effects and these environmental effects that get intertwined. They're difficult to tell apart. I have some ideas about how they relate to each other and how come they are so intertwined and how we can tell them apart. But before I go into that, I think we have to talk for a while about the historical context, about social stratification and the way that we study its relationship with genetics. It's a very complicated history, troublesome and controversial history sometimes. So we're going to go through that first, and then we'll get to the science later. So what is social stratification? It's the way that human civilizations have always stratified their population. So socioeconomic status is the level of wealth, health, power, freedom, overall quality of life. And a human civilization is characterized by, so the definition of a civilization is a society that has some central government, some form of symbolic communication like writing, urbanization, and social stratification. So it's one of the defining characteristics of a human civilization, but not of all societies. So the hunter-gatherer societies that are around today are largely egalitarian, and so anthropologists think that this has been the case in earlier times as well. So of course there are some status differences, but it's not as pronounced as after the agricultural revolution about 12,000 years ago, where food became in abundance, so the population sizes started to increase, and you got urbanization. People had time to develop job specializations and division of labor, so you had different levels of prestige for different jobs and an unequal accumulation of possessions. So there have been many different forms of social stratification throughout the ages. A lot of what I'm going to be discussing applies to the so-called meritocratic system, where people are rewarded a higher status based on their merit, based on what they can contribute to a society. And the earliest recorded example of such a system was in ancient China, where Confucius advocated that those who govern should do so because of merit and not because of inherited status. And in this artwork, you see this old painting, you see people taken a standardized test to see whether they were fit to serve in a governing role. So in Europe, you had this medieval estate system in the Middle Ages, where you had the monarchy on top, and then the religious clergy that gave the mandate to the monarchy, and the nobles and the peasants. And then during the Enlightenment, partly inspired by those old Confucian texts that were rediscovered, the Enlightenment scholars helped introduce the more merit-based system. So the term meritocracy wasn't coined until less than a century ago by Michael Young in his book, The Rise of the Meritocracy. And he did not mean meritocracy as a positive concept when he wrote this book. This book was about a dystopian future society where you had an aristocracy of birth that turned into an aristocracy of talent with a disenfranchised lower class of the less merited. So you had just a new form of social stratification, which was maybe even harder to overcome. And this was a piece that he wrote because he was not happy that meritocracy was turned into a positive concept. He wrote this in 2001. It says, the man who coined the word 4 decades ago wishes Tony Blair would stop using it. I have been sadly disappointed by my 1958 book, The Rise of the Meritocracy. I coined a word which has gone into general circulation, especially in the United States, and most recently found a prominent place in the speeches of Mr. Blair. So if behaviors that are associated with merit, things like intelligence and persistence, if those are heritable, then an aristocracy of talent could theoretically turn back into an aristocracy of birth again. I think that was like behind what Michael Young was trying to convey with this book. And the first law of behavior genetics states that all human behavioral traits are heritable. So then we get into the relationship with genetics. So a trait is said to be heritable if individual differences are attributable to genetic differences. So it doesn't say something about how much genetics are contributing to a trait. It says something about the variation in a population. So it's a relative measure. And the development of the concept of biological heredity, so in the 16th century, it was based on legal concepts of inheritance of property and wealth. And so this concept was further developed and crystallized by people like Gregor Mendel and Charles Darwin in the centuries that followed. And then you had this guy, Francis Galton. He was the cousin of Charles Darwin. And he wrote his book in 1869, where he called Hereditary Genius, where he applied statistics to show that offspring of eminent figures were more likely to succeed in high-profile professions. So we see a family tree here with the Galton part of the family, Darwin and Hedgewood. I think that's the successful businessman that was also related to them. So does anyone see something peculiar about this family tree, something remarkable? So he defined people as brilliant or with scientific ability. Is there something remarkable about this? Yes. So I don't know how much of it is because he was a product of his time or he was an actual bad person, but he was not only sexist, he was also very racist. There are some really horrible passages in this book of his. But he was also objectively a very smart guy. He invented things that we still use to this day in our everyday statistical analysis, things like the correlation, the regression. But he also invented something we are not so happy with today, namely eugenics. So the idea existed before. I think Plato also suggested something like this, but Galton gave it the name eugenics and gave it the credibility, the scientific credibility. He figured that if traits that we value as a society are heritable, then we could potentially influence the genetic quality of a population through selective parenthood. So eugenics was wildly popular in his time, and it was supported across the world, across the political spectrum, from left to right, by established intellectuals and medical authorities, and it was applied, which led to state-sponsored violence on marginalized groups. So things like forced sterilizations. And then during the Second World War, it got linked with race and reached genocidal levels. And one of the arguments of Nazis in the Nuremberg trials were we were just applying these eugenics laws just like they are doing in the US, in the UK. So after the war, popularity of eugenics declined. The science of heredity became decoupled from social applications and progressed through many decades of twin studies. So this slide from this paper summarizes 50 years worth of twin studies. So in twin studies, we compare identical twins with fraternal twins, and if identical twins resemble each other more for a trait, the trait is said to have a heritable component. And almost all of the traits that we looked at, we saw stronger resemblance in identical twins. That's where that first law of behavior genetics comes from, that all behaviors are heritable. And you see the type of traits that were studied. So by far, psychiatric disorders have had the most attention. It's the most mysterious disorder from a biological point of view, and it has a clear genetic component, so it makes sense to try and understand that starting from the DNA sequence. But the first twin study was done on intelligence, and that's one of the strongest correlates with socioeconomic status, if not the strongest in modern societies. This is a review. This comes from a review of familial studies that looked at the resemblance between families and it's ordered by how genetically similar these relatives are. So on top you have the identical twins, below that identical twins reared apart, and then it goes down to dizygotic twins and siblings, and then all the way at the bottom you have the spouse similarities or assortative mating. And we saw already in a couple of talks yesterday that those are significantly higher than 0. And so each dot is a study, and you see that clearly the more genetically similar two relatives are, the higher the correlation between them for intelligence. So these heritability estimates of intelligence are quite high in twin studies, around 80% in some studies. But it's, as I said earlier, heritability is a relative measure, and it depends on the population that you look at, and even which part of the population. So these two studies claimed that the heritability of intelligence depends on the quality of the environment in which kids are raised. So they compared richer families with poorer families, and they saw that the heritability was a lot higher in richer families. So, and they argued that richer families, they have the resources to stimulate and develop the genetic potential of the kids more, so that it makes the heritability go up. These findings are a little bit controversial because they have been difficult to replicate in non-US data sets. I think so. This is a replication but partly the same team. But in Australia and Europe, they did not find this effect. It's not sure whether that has something to do with inequality in the US versus these other continents or the data. But this meta-analysis here is on educational attainment, which is one of the main proxies for socioeconomic status, educational attainment and income, basically. And these are a whole bunch of twin studies done throughout the past century, and you see the heritability change. And the most interesting change, I think, you see in the females, where in the beginning of the previous century, this heritability was not significantly different from 0. So all the differences in educational attainment could be explained by environmental differences, and this was a time where it was not normal for women to go to school. And then as opportunities became more equal throughout the century, you see the heritability of educational attainment increase. So these were twin studies. We are now in a new era, the era of the genome-wide association study. It's a very nice hypothesis-free study design where you measure as many genetic variants as you can in the millions and a very large data set, and you just test every possible genetic variant. And this has taught us a lot about what kind of variants explain these heritability estimates, or at least part of it. And what we learned is that there are many genetic variants that influence the complex traits with very small effects. So you need huge sample sizes. So we have seen these sample sizes increase. And in the past 5 years, they have more than tripled. So the genetic signal has become stronger, but it's still quite a mystery what's inside of these signals. So I said at the beginning that I like to look at the larger patterns of variation in genetic data sets. So my PhD student, David van den Berg, he downloaded all of the GWAS summary statistics from the GWAS catalog and then filtered them to only have big GWASs larger than 50K to have enough statistical power. And then he made this, he was left with about 600 to 700 different traits. And he made a genetic correlation matrix where he computed the genetic correlation between each pair of these traits, and then did the principal component analysis. And so these are the principal components that explain most of the variation. And the one on top, we coined cognition and SES, explains 10% of the variance. So this is a genetic signal that seems to be pervasive in a lot of the GWASs that we do. And that's basically this signal, educational attainment, but also income. These are the main proxies for socioeconomic status in the modern societies, and these two traits, they have a genetic correlation of more than 0.9 with each other. So you capture almost the same trait if you do a GWAS on income or an educational attainment. So what's in this signal? There are the genetic variants don't encode for your high school diploma or the number on your bank account. They influence very low-level basal biological processes, and these influence traits, and these traits influence each other. And they also influence what environment you get exposed to, and that environment influences you, and those traits and biological processes, and all of this gets intertwined in that GWAS signal. So how do you dissect this? How do you tell which correlated traits and environmental influences are in there? Well, we can compute genetic correlations. So here are genetic correlations computed with LD score regression. It's a very elegant method that allows the polygenic signals of different GWASs to be compared with each other. And the highest genetic correlation was observed for IQ. And you see interesting associations with personality dimensions, and a lot with physical and mental health, of course. Normally, the physical and mental health outcomes are often mostly negative, but for the psychiatric disorders, there are a couple of interesting exceptions, namely anorexia, bipolar disorder, and autism. So a positive genetic correlation with educational attainment. And what we did here is we took that educational attainment GWAS and then subtracted the intelligence signal out of it. So intelligence made up about half of the signal of the educational attainment signal. And then you have 50% of other stuff that we call non-cognitive skills. And when you then compute genetic correlations with these components, you see some interesting things happening. You see the intelligence, for example, correlating negatively with bipolar disorder and schizophrenia, but the rest, the non-cognitive skill, correlate positively with schizophrenia and bipolar disorder. So we're not entirely sure yet what this is, but we suspect it might have something to do with openness and creativity. There was another study we published a while ago where we used polygenic scores for bipolar and schizophrenia to predict whether people got a creative job later, and it worked in the Netherlands and in Iceland and in Sweden. So there's still a lot to do there to figure out what traits are contributing to socioeconomic status in modern societies, but we get an idea. And this paper that I started out with, this gave us, this taught us about the relationship with the environment. So as I said, we didn't mean even to write a paper about socioeconomic status. We just, inspired by the GWAS design, wanted to do like a hypothesis-free scan of the geographic patterns of polygenic scores for a wide range of traits. So we made it for 33 traits: the physical, mental health outcomes, substance use, personality, behavior, you name it. So on the x-axis, you see the Moran's I, which is another name for it is spatial autocorrelation. It's a measure of how geographically clustered these polygenic scores are. So how much do neighbors look alike? And before controlling for ancestry differences, almost all of the polygenic scores shows a significant level of geographic clustering. And if you mapped them, you saw maps that looked very similar to those principal components in UK Biobank. So here is the first three principal components. And you see here also sort of the cultural barriers, so the borders between Wales, Scotland, and England. And so we are not sure why a lot of these polygenic scores show similar distributions. It could be real differences. It could also be that their GWASs did not control well enough for population stratification. We saw a bipolar disorder, for example, a map that looked a lot like the second PC with Wales, very red. But we also know that a lot of the bipolar cases in the GWAS came from Bristol, which was in Wales. And if you don't control well for population stratification, then you will see ancestry differences in your signal. So we were not interested in answering that question. We wanted to look beyond ancestry. So we controlled for 100 principal components. We regressed them out of the polygenic scores. The majority of the first 100 showed also significant geographic clustering. So we were a bit stringent with 100. Actually, it didn't matter beyond number 25 approximately, but because we were interested in geography, which is so strongly associated with ancestry, we were tried to be very stringent, and all of the geographic clustering dropped for all of the traits except for one outlier, and that was educational attainment. And as I said earlier, if you plot those on a map, you see these maps of socioeconomic differences. Those black lines are coal mining regions. The coal mining industry collapsed somewhere between the 20s and the 80s in the previous century. There was a lot of economic problems back then, a lot of joblessness and other environmental stressors that made people want to get out of there. And we saw that the people that were born there and were able to migrate out of the region had a higher polygenic score than the entire country on average. So the regional differences seemed to be driven by a brain drain that was detectable on a genetic level. So you see in the left graph there, you see the blue line was people born outside of the coal mining region, but you see the other one, the red line, dropping much faster, or significantly faster, and that was because of, in the next plot, we split them up further. The green line that's on top, that's consistently higher than the rest of the country throughout the century, that came from those coal mining regions. So that's why you see these differences growing. And another analysis that showed what was happening in a different way was when we compared birthplace with current address. So here on the x-axis you see another measure for geographic clustering, and you see that for educational attainment, current address shows stronger geographic clustering than the birthplace. So that's because of the migration is increasing this geographic clustering. But for those principal components that reflect these older ancestry differences, you see the other way around. You see that migration is breaking down those old geographic patterns. So the geographic clustering decreases from birthplace to current address. So you have these very old, ancient ancestry differences that show these old geographic patterns. Those get broken down by that more recent migration that's driven by socioeconomic status, and that more recent migration is increasing the clustering of the polygenic scores. So this is a preprint that came out recently where they did the same analysis in Estonian biobank. So the Estonian biobank genotyped about 20% of their population, about 180,000 people. And they looked at way more polygenic scores than we did. We looked only at 33 polygenic scores. It was because at the time there were few large GWASs available where UK Biobank was not the majority of the sample. But they did not have this problem in Estonia, so they looked at 150 or 160 traits, and they saw the same outlier. Educational attainment showed the strongest geographic clustering, and they saw also, if you compare birthplace with current address, that current address shows stronger geographic clustering, and for their principal components, they also saw it the other way around. So you have here those maps show those old ancestry differences in Estonia. Those get broken down by migration, and instead you get this geographic clustering of the polygenic score of educational attainment, and the map on top, and there you see these two university cities light up. So the brain drain is because of the higher polygenic scores that are migrating to Tartu and Tallinn. And on the left graph, so on the x-axis, you see the genetic correlation of those traits with educational attainment. So also what you see there is that for the other traits, the higher their genetic correlation is with educational attainment, the stronger the geographic clustering is. And we saw the same thing also in UK Biobank. On the x-axis, you have that Moran's I again, and on the y-axis, the genetic correlation with educational attainment, with intelligence there showing the highest genetic correlation with educational attainment and the highest level of geographic clustering. And so educational attainment would have a Moran's I of about 0.6 and a genetic correlation of 1, so that would be all the way on the right top of the slide. And so it seems as if educational attainment is pulling all of these other traits with it geographically. And it's not entirely clear how much of that is because of environmental influences, because this geographic clustering is not random. It's clustering with these environmental differences. It's very different to live in a poor region than in a rich region, especially in the UK. It's the most unequal country in Northern Europe, with I think the 5 poorest cities and the richest region as well, all in the UK. And this is a map that's just from census data of a clearly environmental variable, namely the number of fast food restaurants in your area. And that shows the same map as that polygenic score. So there's a lot of information on this slide, but let's focus on height, the phenotype height. So you see there those crosses are the polygenic scores of 4 traits. So it's the polygenic score for educational attainment that is explaining 1% of the individual differences in height. And then you have that green cross explaining 21% of the individual differences in height. So that's. So height has a very strong polygenic score, and it's a very heritable trait, about 80%. But what happens if you take the regional average of the polygenic score and the regional average of height, then suddenly you see that the polygenic score of educational attainment outperforms the polygenic score of height in predicting these regional differences in height. And that is because this educational attainment polygenic score on a regional level is a proxy for these environmental differences. So here you see on the left the distribution of the height polygenic score, and there you see that the people in the coal mining region, if it was 100% determined by genetics, they would be the tallest of the country, but instead they're among the shortest, and that's because of these environmental differences. So here we further explore these gene-environment correlations. There are if you get born into a family where you inherit the genes and the environment from your parents, and those two are correlated. If your parents have a higher polygenic score for educational attainment, it's more likely that they also have better resources to provide for your development. We call that passive gene-environment correlations. But there are also small genetic differences between those siblings. And we saw traces of active gene-environment correlation, where the sibling with the higher polygenic score was more likely to go to a region with better environmental influences. And so you have this, now the way to reduce these gene-environment correlations is by doing a within-family GWAS, where you compare siblings with each other, but that would not control for these active gene-environment correlation effects. And in this paper, we did GWASs on more than 50 different traits, a wide range of physical and mental health outcomes, and then controlled for the region that people live in, birthplace, current address, and both. And we saw that the heritability decreased only for traits that were really closely related to socioeconomic status, so things like educational attainment, income, intelligence, time spent watching TV, overall health, and age at first birth. So these are the traits that are most strongly correlated with socioeconomic status. But if you looked at the genetic correlation with educational attainment, you saw that that decreased for almost all of the traits, and most strongly for things related to BMI and substance use, so things that you put into your body. By controlling where these people were born and where they migrated to, you saw the genetic correlation decrease more strongly even with income, well, income and educational attainment are almost the same signal. So they say that nothing in biology makes sense except in the light of evolution. I think that's even more so with genetics. So Darwin, he wrote that nature selects adaptations that give organisms an advantage in the three struggles of life against physical environment, other species, and members of one's own species. So humans have made great progress conquering the first two since the agricultural revolution, not entirely, of course, but the competition against your own is even increased, I would say, where if you lose that competition, you get a lower SES, worse environment with more higher disease risk and higher mortality rates. So what I did here, so when the COVID-19 pandemic broke out, you could every day you could download the new reported cases and deaths on a regional level from the UK. And I know from UK Biobank where everyone lives. So I downloaded the reported cases, but also the deaths on a regional level, and then linked this to UK Biobank participants, and then did a GWAS, which we call a regional GWAS, on the amount of COVID-19 cases in your region. And then computed the genetic correlation with educational attainment. So each dot is a GWAS here. And you see that in the beginning of the pandemic, the first day, there was a very, very strong genetic correlation with educational attainment, but it was positive because the richer region of the country, that's where the virus entered the country to tourists, probably, or people that travel there, and then it very quickly drops and becomes negative because it spreads to the poorer parts of the country. You see one spike there in December, that's because of that new, more contagious variant that came into the country, but that also quickly drops. So COVID-19 rates they target the same genes that are targeted by social stratification, by socioeconomic status. But COVID-19 is not likely to exert a strong selection pressure because it's most deadly for people beyond the reproductive age. But previous and more deadly pandemics showed the same patterns throughout history. So these unhealthy living circumstances that we talked about, together with these regularly occurring pandemics, especially since the advent of the agriculture, where people were living more packed together and with other animals. So you can conceive that if social stratification targets the same traits, that this could be a selection pressure that changes the allele frequencies in the population. But natural selection doesn't only occur through mortality rates, it occurs through differential reproductive rates, which are influenced by both mortality and fecundity, number of children people have, and mate choice. So in this article, we looked at these same 30 polygenic scores, but associated with the number of children. And here also we saw educational attainment showed the strongest association with number of children. And for the other traits, their association with number of children was very dependent on their genetic correlation with educational attainment. Just like we saw with geographic clustering, it seems that educational attainment is driving all of the other associations as well. Except this was in the opposite direction as with the mortality rates, where we saw higher polygenic scores actually having fewer kids. But this depended also on the part of the sample that you looked at. So here we could also only do this for the mothers. Split it up into mothers that had their first child at a younger age, younger than 22 and older than 22. And then for both groups, you see a very quite strong and significant relationship, but in the opposite direction. So mothers that have their children at age 22 or higher have a strong positive association between educational attainment, polygenic score, and number of children. It was written with an economist, David Hugh Jones. He had the old economy hypothesis where you have two different forces. One's called the substitution effect, where women wait longer to have children if they are pursuing a career, which would cause that negative relationship. And then you have the income effect, where if you make more money, you can afford to have more kids, and you have these two opposing forces. But overall, there was a negative relationship, which is a relatively recent phenomenon. Because so this study looked at wills from before the Industrial Revolution, and there we saw that wealthiest left a lot more offspring. This is another study that looked at more countries over a longer time period, and they saw the same thing, that there was first a positive relationship, and then after the Industrial Revolution, it turned around. There are different explanations for this. Child mortality rates decreased, better birth control, women waiting longer to enter the job market. But in some Scandinavian countries, it's actually turning around again. So it's positive now in a couple of Scandinavian countries. And then you have another process that influences the genetic makeup of population. That's mate choice, assortative mating in particular. It can increase genetic differences between families and decrease them within families. And it also has an effect on our effect estimates in GWASs. I like to compare it with, imagine you have two schools, one in a rich neighborhood and one in a poor neighborhood, and the one in the rich neighborhood is attracting all of the good teachers. If you would test the effect of one teacher in that rich school, it would seem as if he's doing a lot better than he actually is, because he's surrounded by all of these other good teachers as well. Same happens on a genetic level. When you have assortative mating, you combine all of these genetic variants with very small effects in the same people, and then single genetic variant will seem to have a stronger effect than it actually has. So there are a couple of studies that have looked at this. Some of them were presented yesterday, and all of them see strong assortative mating for educational attainment. So here, PhD student of mine, Melanie DeWitt, looked at the same 33 polygenic scores in spouse pairs in UK Biobank, and that dark blue corner there, that's educational attainment, is again the outlier here. And then to make things a little more complicated, so this is a recent working paper led by David Hugh Jones, the economist again, where he looked at an environmental shock, namely birth order. So for some reason, kids that were born earlier in a family have a higher socioeconomic status, but not because of genetic reasons, because on average they have the same genetic makeup as their siblings. But the polygenic score of their spouse pair is also higher. So they're trading socioeconomic status apart. That's the part that is influenced by environmental circumstances for genetic material, and that way that's another pathway where you get these environmental and genetic influences intertwined, entangled in other subsequent generations. So to conclude, socioeconomic status, it's a network of complex traits and environmental circumstances woven together by social and economic forces, and a better understanding of how this works can improve not only studies in the social science genomics area, but all of our GWASs, all the physical and mental health outcomes are affected by these things. I think it may be a dynamic selection pressure. I say dynamic because it changes over time and between societies what traits are valued. So we saw it reverse since the Industrial Revolution, and I can imagine that if there are certain traits that are more consistently advantageous, something like intelligence, that maybe that the selection pressure could have a bigger impact, and that could maybe be part of the explanation why our societies have in such a relatively short time become so ridiculously complex and are increasing to become more complex. And SES, it's also driving migration and assortative mating, which increases inequalities on both an economic and a genetic level, which could make them harder to overcome. And I'm not sure what to do with this information on like a social policy level, because if you try to control the genetic makeup of a population, that's pretty much the definition of eugenics. So I would love to hear your thoughts if you have any. And so I did not do this work alone. I got a lot of help from all of these very smart people on this slide. So thank you very much to all my colleagues and also to you for your attention. We have, I think, 10 minutes, right, for questions?
This is fascinating. Thank you. Loved it. So I love your results on the geography, but I can't say I was surprised by them, right? So as an economist, there's a huge, and in social sciences, there's a huge literature on migration and what make people migrate and immigrants being positively selected, and Latin immigrants in the U.S. are healthier. So I think it's fantastic that we see that in genes, but I wasn't surprised that it was also the EA and not some of these other traits, right? And so I think my first question is, were you surprised, or was that your hypothesis when you started looking at these problems? And then to kind of come back to your last question, I mean, if it's people migrating voluntarily for a better life,
Mm-hmm.
I don't think we should do anything about it, right?
Yeah.
And okay, one more question. Are you I think if say you had the data to look at Mexico, US, I think you would see even more stronger patterns, right? Because there the barriers are higher, so you would see even more selection, I would think. So, yeah.
Okay.
Just
Thank
kind
you.
of like comments more than,
Yeah.
but it's fascinating.
Yeah, no, but those are very good questions, and questions I ask myself. Well, your first question was I surprised? I was, because I didn't have an hypothesis. I just was looking at everything. I didn't know about the coal mining regions. When I looked at the map, like I had a colleague, David Hugh Jones, who is an economist, who recognized the coal mining regions.
Mm-hmm.
And I was surprised that it was just educational attainment that was seeming to drive all of the others. It's not something I expected. Your second question was?
Should we do it? I don't know. Is there anything to be done?
Yes.
If it's just
Yeah, I don't know. Like we see, it's a logical conclusion that these processes are increasing inequality on both an economic level. That's not a surprise, but maybe also on a genetic, partly biological level.
right.
And I think you don't need genetics to come to this conclusion, but I think one of the things that we maybe could think about in society is that why is that this punishment for not being able to climb that ladder should maybe not be so severe.
If you go back to the Mexico, right? That is, should we not allow like extremely smart immigrants to come to this country?
Yeah.
Because of the increase, I think it's a bigger question, right?
Yeah,
Because
so
of the bigger inequality, but there's also bigger growth, maybe, right?
Yeah.
Yeah.
Yeah, so you're now talking about like between countries. So I specifically focused only on internal migration and also people of the same ethnicity. But between countries, I mean, this brain drain could have, like, it's good for the country that gets all the brains, but not so much for the areas that are left behind. And I'm not a sociologist or social policy expert, so I don't know. I look at the data and I see these effects, and you see a trend that's increasing differences, and increased inequality is just associated with many bad things like social unrest and it's health differences, and yeah, it's something we should think about, but I can't do that. That needs multiple disciplines to put their brains together and think about this. Yeah.
Thank you.
Thanks.
Thanks very much for the talk. I have two questions. The first is, sometimes we get pushbacks from the human genetics community about things like GWAS and education attainment. One of the arguments would be that we did a particularly bad job when estimating the SNP effect on socioeconomic outcomes because these SNP level estimates are going to be fundamentally confounding. So I wonder if someone says, looks at this result and comment that the correlation between EAPGS with geography reflects confounding we didn't correctly remove in GWAS. What's the single piece of evidence you would point out to counter that point? It's the other way around. And the second question is, I think a lot of us feel the need to eventually generalize this type of work to more diverse populations. We're very familiar with the convention that people just use non-Hispanic white population in a biobank to do GWAS like this. But I wonder if you can comment on the current fundamental challenges if we were to do this today and involve everybody in work like this, what would be the hurdles and roadblocks you see?
Yeah. Okay, so I think I'm going to need some clarifications for your second question, but let me try to answer your first question first. So people will rightly say these polygenic scores are highly confounded, and how do you know you're not just looking at all environmental influences? Well, we have certain approaches like LD score regression that can tell apart actual polygenic effects from confounding. We have the within family and within geography designs that show that a big part and can quantify how much of the genetic signal is due to inflation because of environment or assortative mating. So so people that criticize these GWASs and say you're just picking up confounding, it makes this trait, these proxies for socioeconomic status, the most difficult traits to study, but I also think the most interesting because there's a lot going on in there, and I find it very interesting to dissect, well, how much is because of the assortative mating, how much is because of the environment. And you have different designs that allow us to follow up those effect estimates and try to dissect what is causing what, and that's also teaching us something not only about the genetic effects but also about how our society works and how these effects differ between societies. So that would be my answer to that criticism. And for the second question, I wasn't sure whether, do you mean like if we would diversify our data sets?
Let's say we have the data, right?
Yeah.
Ancestrally diverse. Is
Yeah.
there a technical reason or ethical reason, whatever reason, that is stopping us from doing this type of work today? Just generalize exact same methodology to a diverse cohort.
Yeah, so this relates to your first question as well. All these confounders, we don't even understand them well enough within populations, but these environmental differences and socioeconomic differences are much stronger between populations, even between ethnicities in the same country. It's different to grow up as, like, let's say a Moroccan in the Netherlands. I was born in the Netherlands, but with Moroccan ancestry. I grew up in a different environment than someone with the Dutch ancestry. And so the type of confounding that makes it difficult to study these traits within a population will make it even more different between between populations. The differences between countries are again from a different order. So I tried to focus on this first and try to figure this out. But at some point we'll get there and have to combine all these more diverse data sets and try to understand this complex relationship between culture, socioeconomic systems, and genetics. I think we'll get there, but it will be very difficult, and we're not even done with understanding fully what's happening within these ancestries.
Maybe one more. Great talk. Just a quick question. Maybe I might have missed it, but given that we now have like more sample, like within family sample, if we estimate the PGIs or the PGAs using within family sample and get closer to estimating the direct genetic effects of like, for instance, EA, so would you think the results would change? Because if we get to the more direct estimate of like maybe less biased estimate of educational attainment, would you think like all these results would change given that we got to estimate like EA more effectively?
Yeah, so that's a good question, but also, like, I'm not sure how to get to like the exact. I don't think within family is enough to remove everything, because we also saw these active gene environment correlations that we see when we use geographic region, and geographic region is only a rough proxy of your social environment. Your social environment is something that's very difficult to measure and that changes throughout your life. So I think it's a little misleading to say that within families, GWAS captures direct genetic effects. I don't know if we will ever be able to measure the environment well enough to remove all of these intertwined gene environment effects. Yeah, and I think your question was, will the results change if we would hypothetically have them? Potentially, yeah. Like some of the results I saw for other traits were driven by their genetic correlation with educational attainment, for example. I don't know how much of that genetic correlation is because of environmental influences or because of pleiotropy, or that could become more clear if we have more direct estimates, but that's very difficult to achieve, I think.
Yeah, we're in the freeze. Yeah, because no more Chinese students. So the universities.
All right, welcome back. Our next session is on gene-environment interaction methods. G by E is really, really hard to study, measure, quantify, estimate, characterize, whatever you want to say. It's a notoriously sticky issue. So let's hear about some of the really cool and innovative ways that scientists today have been tackling that particular issue of measurement and studying G by E effects, starting with Wenmin Zhang.
Thank you for the nice introduction. I'm Wenmin from a postdoc from Montreal Heart Institute. So I'm a data scientist by training, and I mostly work with graphical models where we use nodes and edges to represent the relationship between different variables and find efficient algorithm to study about them. Today I'm going to talk about one of them that can improve power for G by E analysis. I'll begin with the key idea behind the method and then share some real data examples, and in the end conclude with a brief discussion. So first, we all know that G by E analysis is very important. In the context of medical research, mostly for identifying modifiable risk factors and protecting susceptible individuals. Usually it's done with regression by including the genetic variants, the environment factors, as well as a G by E term in a regression model, one variant at a time. So this means that you will have a very high multiple testing burden. So here is a simulated example to illustrate this. There is one causal variant having different effect size in two populations in this, so that when we perform exposure stratified GWAS, we can see a very different pattern here. However, if we are going to test G by E on this example, we'll get a very noisy signal. And after multiple testing correction, we got nothing. So is there a better way to do this? If we know the causal variant, it will be quite straightforward. Given the genotype of the causal variant and the phenotypes in two different populations, we can estimate the effect size of this causal variant in different populations. And then simply by comparing the effect sizes, we can detect the GWAS interaction effect. However, in reality, we almost never know the causal variant. And this is itself a quite challenging question in statistical genetics called fine mapping, mostly because of a phenomenon called linkage disequilibrium, where variants in close proximity to each other often travel together. So here we are visualizing the pairwise correlation between different variants, and a red block here represents a group of variants in high correlation with each other. So the approach we took to tackle this is introducing latent variables called effect groups, where we are grouping the correlative variables together. So ideally, we want to have a causal variant and the other variants in highly correlated with it in the effect groups. And then we use the effect groups to align the causal signals across different populations. Therefore, we can estimate whether the causal status of the effect groups in different populations, as well as the effect size, so that we can compare them. And now, by performing the GWAS analysis at the effect group level, we can greatly reduce the multiple testing burden for GWAS analysis. So this is the key idea behind our method called SharePro. So here we take the genotype and the phenotype of the in different populations as input, or alternatively the GWAS summer statistics in different populations as input, and infer the composition of the effect groups as well as the causal status of the effect groups in different populations and and their causal effect sizes. So the green shaded variables are what we observe, and the white, orange unshaded nodes are what we want to learn about. So this is what we call the probabilistic graphical model, where each nodes are variables and they have probability distribution associated with them. And now our goal is to infer the distribution of the latent variables given the observed variables. So this can be done through the Bayes theorem. And here the numerator is very easy to compute based on our model assumptions. But the denominator is actually very hard to compute. So we use a technique called variational inference. Basically, we approximate the hard to compute complicated posterior distribution with something we can easily manipulate called variational distributions by minimizing their difference, so that we can have a very efficient algorithm to detect the effect groups for GWAS analysis. So in the previous simulated example, sorry, how do you go back? So in the previous simulated example, we can see that taking the exposure stratified GWAS as input, we can identify one effect groups consist of two variants in high LD with each other using SharePro. And by detecting GWAS analysis in this locus, we can successfully identify the simulated effect heterogeneity without worrying about the multiple testing correction. So there are a lot of simulations, but they always work as expected, so I will skip them. So let's look at some real data examples. The first one is GWAS smoking interaction effect on lung function. indicated by the FFR. We used the UK Biobank to perform the smoking status stratified GWAS, and then perform GWAS analysis comparing different groups, the current smoker, past smoker, and never smoker. And this is the GWAS result. The only signal at the bone-foramen threshold is the map to the CHRNA3 gene, which is the famous smoking gene. And using SharePro, we were able to identify around 300 effect groups. And not only did we identify the CHRNA3 signal, we also identified other genes that map to biological functions, including airway inflammation and the DNA repair. So this is the power of SharePro. And then alternatively, people sometimes using the meta-analysis results to identify candidate variants for GWAS analysis. And if you do this in this case, you'll miss all the GWAS signals. So sometimes it's not a good idea to do that. And the next example is the sex-differentiated genetic effect on fat distribution. This time, our goal is to find the mediating factors that can be important in shaping the sex difference. Again, we used SharePro on sex-stratified GWAS on waist-hip ratio adjusted for BMI, and we identified around 400 effect groups. So here, each dot is an effect groups. And the first candidate we look is the sex hormone binding protein. So in the x-axis, we're plotting the sex-differentiated effect on sex hormone binding protein against the sex-differentiated effect on waist-hip ratio. So we can see a very strong correlation here. And in contrast, when we look at testosterone level, there's not a significant correlation. So that means that probably the female hormone might play a more important role in shaping the sex difference. Additionally, we looked at the 3 lipids level, including triglycerides, LDL, and HDL. So among them, triglyceride is the one that mostly directly related with diet. So as a result, it has the strongest correlation. So in summary, variant level GWAS analysis can have a very high multiple testing burden, and SharePro can reduce the multiple testing burden through joint analysis, the exposure stratified GWAS, and SharePro achieved a higher power than the variant level GWAS analysis, and potentially it can be useful for identifying the mediating pathways for shaping the GWAS difference. And the software is openly available on GitHub, and all the codes to reproduce the analysis is available by scanning the QR code. And in the end, I would like to thank you, my supervisors and funding agencies. Questions?
Thank you for the talk. In general, statistical inference after variable selection is considered a difficult problem, but that sounds like what you're doing, right? You generate a hypothesis and shrink down the number of hypotheses, then perform hypothesis testing and calculate p-value from the same data. I wonder if you can comment on the risk of double dipping, because can you ensure the p-values after this is still valid?
So actually, the two process is different. So in the fine mapping process, we are only looking for, So in the fine mapping process, Sorry, it's too sensitive. So in the fine mapping process, we are looking for the signals that are significant in either exposure groups. And then in the GWAS stage, we are actually testing the difference. So it's two different analyses. So it has a relatively minimal risk of double dipping.
No one else is asking question. I'll just ask a follow-up.
Yeah.
So then a simpler approach is to simply do clumping, right, for each respective GWAS, and then select top variant based on p-value.
Yeah. This is the result for clumping.
Do you have intuition why the power is completely gone?
Because clumping is usually performed on the meta-analysis of GWAS or the main.
No, but you can do
backbone.
clumping for each, for example, sex stratified GWAS.
Yeah.
Right?
So when you stratify do stratify analysis, you have smaller power, a smaller sample size in each group. So that means that you will lose the power to detect the signal independently. But then here we are actually jointly analysis across all the groups, so you can maintain the signals in either population. So you're actually borrowing sample from the other exposures. Yeah.
Thank you.
Any other questions? Okay, thank you. Sorry.
I think it's a quick question. Could you just, so you were using biobank scale examples.
Yeah.
How small of a sample can you have and still use your approach?
So it depends on how noisy is the background. So in general, so during the inference stage, we're actually looking for signals that stands out from the background. So if the effect size is relatively large, then you don't really need a very large sample size. But if it's and then you need a very large sample size.
So ApoE might be good, and the smoking one might be, but there's a lot that would be too small. Is that?
So the purpose of this method is to increase the power for GWAS analysis. So the ones that you missed in previous analysis might able to be identified through this approach. Yeah.
Is that the actual last question? Okay.
Is this a microphone as well? Is this a microphone as well? Here. Okay. Hi everyone, I am Marina. I'm a postdoctoral student at the Vrije Universiteit in Amsterdam. I'm going to be presenting some very preliminary work for you guys today. It's work.
Could you stay either with the mic or keep the mic with you?
Okay, sorry. Yeah, I have problems with staying put, so follow me around. So yeah, today I'm going to be presenting very preliminary work, and it's work that's been done jointly with Sylvia and Leandro, who are over there, and also Titus and Sjoerd, who are not here, but many of you know, I'm sure. So the work is around Alzheimer's disease and related dementia. And obviously, it is a growing problem with the population becoming older. And global prevalence is projected to be around 66 million people by 2030. There are high direct and indirect costs associated with ADRD. And for policy purpose, it is important to understand more about these costs. And that's what we're going to do today. So our contribution to this literature is we have very detailed data, not only on genetics, but on a registry of everyone living with dementia in the Netherlands. And also, we joined that with the universe of taxpayers in the Netherlands. So joining all these different data sets to understand the socioeconomic costs involved in ADRD. Just to tell you again, because of the big effort in terms of data analysis, what I'm going to be presenting is still quite preliminary, and we still I told many people, which is a sort of a metaphor that I like, we're still in the prepping phase of our kitchen. So I'm going to show you the data kitchen right now. I can't show you the prepared meal yet, so okay, bear with me for this presentation. So what are you going to see today? I think one of the first results that we want to see as geneticists is that genetic variables do matter for ADRD, and we do find that, of course. Another thing that Sylvia presented yesterday, and that we also find in the Netherlands, is that there is little evidence of environmental confounding with when we control for parental genetics. Another thing, there are many problems with using these large data sets for, well, not using the data sets, with researching ADRD. One of them is like people self-select. The diagnosis is not exogenous, right? And another problem that we found is selective mortality. So in the high-risk ADRD group, we see that only, well, the relatively highly educated survive more. So that's a problem that we'd have to tackle as well. And one of the things that we do to tackle this problem is because we're using the universe of taxpayers, we can find their children irrespective of them being alive or not later on. So we're going to focus on the income and wealth trajectories of children, of individuals living with ADRD, okay? So, yeah, I already told you about the data, but I'll tell you again in a little more detail. The genetic data comes from the Lifelines, which is a genotype cohort, a longitudinal cohort of people living in the Northern Netherlands. The other data sets that we're using come from the CBS, which is Statistics Netherlands. And this is a registry-based measure of dementia with age of diagnosis, and diagnosis is happening anywhere between 2006 and 2022. So, and just to clarify, like they come from, this dementia diagnosis comes from various sources where anyone could have received any care or service related to dementia. So it doesn't come only from health facilities where, I don't know, receive medication, or you go to a geriatric specialist or to a mental health specialist, but also from nursing homes, for example, okay? So it's quite a kind of wide data set. Besides that, as Leandro presented early today, we also have the tax registry. So it's the universe of every taxpayer in the Netherlands. It's literally 17 million people that are in the Netherlands. And we link this to the Lifelines. And it has all their data, so on income and wealth. Again, from the years of 2006 to 2022. And from the Lifelines, because we're talking about ADRD, we focus on people who are relatively older, and we focus on a group born before 1967 and who have been genotyped. It's around 35,000 people, okay? A bit of descriptive stats. Because we have so many time periods, I decided to show you, and it's really small, so sorry. But so, for example, if you look at taxable income for 35,000 individuals, we see that on average here is 30,000 euros, more or less, but this has been deflated to reflect 2015 euros, of course, so it will be a bit more today. Again, for brutto income is a bit higher, right? So brutto income is usually higher than taxable income. That's around 36,000. And we go into the children, well, wealth, around 250,000 euros, net wealth. So it can have negative values as well. And when we look at children, and here just to note that the children variables are the average for children. So if you have 3 children, I pick the average for the 3 children and attach that to the parent, okay? So this is average values for children. They are quite a lot poorer than the parents. Some of them might not even be working yet, right? So they earn on average 22,000 euros, which in 2015 value, and net wealth is around half of that, of the parents, okay? Just to bring also some statistics on these parents, the average year of birth is 1955, and they have around 12 to 13 years of education. And of course, as we are talking about ADRD, one of the main variables that we care about is the ApoE. And the ApoE status is consistent with what we see in other samples like the UKB, okay? With most people being the neutral category of ApoE E3, E3. Sorry, did you want to go back?
No.
I don't go forward. Oh. Okay, here. Thanks. He already promised some good feedback, so he's working on it. So here, like our first, what we call our first stage results is just simply using genetic variables to predict dementia diagnosis. And it's a simple model where we use a PGI of CUNCO, we exclude the ApoE region, and then model each ApoE group separately, with the neutral group, E3, E3, being the excluded category in our regression analysis, okay? Again, this is our main specification. We'll try next specification where we also include the parental PGI, using imputing trios. And once we have this specification, one of the things that we do is that we use the predicted value from this equation to construct a composite measure of genetic risk. So here are the results of this analysis. So the first column represents, and again, is maybe a bit small, but a bit better than last time, our preferred specification. Again, we have a important effect for both the CUNCO weight and the ApoE high risk categories, as expected. And one of the things that I'm doing in columns 2 to 4 is basically showing you that parental genes don't really matter. So what we look is like, if we run just a specification with just the CUNCO PGI, we haven't yet computed ApoE regions for the parents and their children. We only did the imputation. We only did it for the PGI. Is that if you go from the second to the third column, we see that controlling for the parental PGI reduces the coefficient quite a lot. But what I'm arguing by showing you the fourth column is that that reduction is mostly to do with the samples that's very different. So if we were to keep the sample the same but running the first regression on column 2, we still get a coefficient that's much closer. So in this, we conclude that, and with other analysis I'm going to show you, that parental PGI doesn't seem to be so predictive of dementia, okay? And because of the sample size difference, we choose to ignore it and focus on the bigger sample. So using our preferred specification, we compute a composite risk of ADRD, and we separate that variable into 3 categories, like terciles. So the top tercile, the middle tercile, and the lower tercile. And we run a Cox hazard model on these risks. So the composite risk has a hazard ratio of 1.3, as expected, and is highly predictive as well of age of diagnosis. And if we separate between the high and the middle group, leaving the low ADRD risk, genetic risk group out, we see that the middle group, we have a hazard ratio of 1.2, and the high group is almost 2, so you're twice as likely to die at a younger age or survive Alzheimer's. Here is one more evidence that the parental PGI isn't confounding on other diseases. So we have a range of diseases for both mother and father. So Parkinson, heart disease, and cancer, any type of cancer. And we showed that the PGI for dementia obviously predicts only dementia. It doesn't predict any other disease. And once we control for the parental PGI, that we no longer can predict parental dementia, right? That makes sense, because it's all driven by the parental PGI. Oh my God, sorry, I have 2 minutes left. Okay, now some of the results, they are still preliminary, as I say, and they're mostly sort of correlational we showed the top and bottom terciles of ADHD risk, and we plot the trajectories of education on brutal income and brutal income of the average child. So here in education, I think there's something that's interesting, is that as the person ages, in the high-risk group, there is a divergence between the high and the low-risk group. That means, was what I said, there is a selective mortality for the high-risk group with only highly educated individuals surviving. And that poses a problem for our strategy. Because these, if we're going to see things going through time, we're going to see two things happening, not only ADHD, but also the fact that the ADHD people that are surviving have higher education. So if we look at income, we see the same pattern, where, well, mostly it's those two lines are on top of each other, and then at the end of life, we see the blue line separating, which means, again, that we don't know if it's happening because these people have higher education on average as well. So for children, we don't see a big effect yet, but I couldn't bring the actual graph to you now. But if we separate this through not only children, but to the average male or female child, we see a much bigger effect for females. Okay, I couldn't bring it yet. So what did we find out today? That genetics is highly predictive for ADHD. There's little evidence of environmental confounding and evidence of selective mortality for individuals in the high ADHD risk group. And we focus on the children to partially eliminate this problem. The next steps is to look at estimated effects by gender, which is brought some sort of new evidence to you on that. And also to link the data to mortality. We also have in the CBS, Statistic Netherlands, the data on mortality to better understand this whole point of selective mortality in the high ADHD versus whole low risk group, sorry. And we're also thinking about new methods to correct for mortality selection, like using individual fixed effects and other kind of methods. Okay, thank you.
So great talk. Thank you so much. Is this on? Okay, it is.
Yeah.
So I'm just wondering about your thoughts about the predictive utility of variants beyond ApoE for dementia. Just your general thoughts about that issue.
I think it is an issue that we already touched here in the presentation. So we had an earlier presentation on which PGIs to use, and it's clear that AD by proxy is not a good PGI, so that's why we chose the Kanko PGI. It does much better at predicting AD, but in our case, it's actually quite predictive using that PGI. It is, and even if you control for ApoE, I mean, we use the PGI, not the total PGI, because the total PGI does have the ApoE region, right? But we exclude the ApoE region and model it separately, because we know from previous studies that ApoE has a nonlinear effect on the ADHD risk, right? With the top A3 category increasing risk by almost 15-fold. So, and even though we're controlling for both the ApoE risk, the PGI is still highly predictive of diagnosis in this case.
So like how much R-squared do you pick up? Just roughly?
Oh, the R-squared is really small still, so it's about 0.5%.
Yeah,
even
okay,
with,
but beyond the ApoE region.
Yeah, it would be really, really like less than 0.5%. Like
Okay, but there is predictive validity beyond the ApoE region.
there is predictive. I mean, if you look at the significance, is also predictive in a sense. I mean, small effect, but it's significant. So
Yeah, thank you.
yeah, okay.
And as people age more, right, this R square gets higher, but as you have more variance. Well, we can talk, yeah, sorry. We can talk.
Yeah.
Thank you. I think this is super interesting data and connecting it with the children. I guess, like for me, knowing it kind of takes a lifetime really to develop this AD pathology, unless you have really early onset Alzheimer's disease. What was kind of your hypothesis in thinking you might see it in the children? Or why did you think that that might be, that you might be able
Oh
to
no, the hypothesis we're having is that basically when your father or mother develops ADRD, you're going to leave the labor market and your income is going to decrease,
Okay.
especially for women who are
So it's looking at how it's affecting
the whole household. So you have direct costs, though, I mean directly into the own individual, and you
Okay.
have costs in the whole family for ADRD.
Okay.
It's
but
more
you're not
around
seeing
that.
those income effects in the children.
We're not seeing average effects, but when we look by, I couldn't bring it, I was literally running the stuff, and you can't access the CBS data set from the US. So I couldn't export it on time to bring it here, but if you look by gender, then you start to see things.
Okay, I see. Okay, thank you for
Yeah.
clarifying.
Costs of informal care, right? That's what we're trying to get.
diagnosed? Do you know
Yeah, we're gonna use, in the next step, we're gonna use the date of diagnosis also.
Okay, so now.
But of course, the cost might come before diagnosis, right?
Right.
Yeah.
Any other questions? One more question.
Thanks for the talk, Marina. So I don't know much about Alzheimer's. I know a lot more about depression. And we know that in depression, there are sometimes effects from the younger generation to the older generation. So if your kids are particularly troubled, that might lead to depression onset. Is there such a concern in Alzheimer's at all? Have you looked at?
I think because Alzheimer's is mostly happens during later in life, then it would be mostly the elderly kind of posing problems to the younger member of the families. But we are also, I mean, we haven't brought it, but we could also look at spouse effects, right? So in that sense, same age effects. Is that?
But what if your middle adulthood was really stressful because you had a really difficult child, for example? Would that affect your risk of developing Alzheimer's at all?
I don't know, to be honest. Like this question, yeah. I mean, there's obviously like education is a risk, protective factor, and so I don't know if some of this happens for socioeconomic status as well, which then would be related to things like how hard your childhood was. So yeah, but I don't know the exact precise estimates.
Thank you.
Thank you.
We're going to do
I have to stay.
a cup change first.
Oh, sorry. It's yours, okay. Sorry.
Which is the thing that I walk around with? This guy?
Yeah, you need to. Yeah. If you like to walk around like me.
Okay. Oh,
This is
guy.
this
Am
is
I-
the one
okay,
that changes.
beautiful.
That one is a microphone.
Excellent. Okay,
Yeah.
beautiful. Look at that. Okay, I'm getting there. All right, thanks. And so this paper is going to be a little bit different. Today I'm going to be wearing my applied econometrics hat. So I sort of view this paper as sort of being a nice companion to Anya's keynote yesterday, in the sense that I think that one of the lessons from this paper is going to basically overlap with hers, and you'll see that in about 12 minutes. This is joint work with Wei Li Ding, my colleague at Queen's, as well as Genia Lukanova, who, if she was here, would fit the all postdoc theme. I'm no longer a postdoc. Okay, so, yeah, so, okay, we'll get there. All right, so a little bit of polygenic scores, are they overhyped or not? And so a lot of this is going to come from a different place. So whereas Anya was thinking about everything from ethics and legal perspective, anyone who knows anything about nutritionists, they don't care about ethics or legal things, so this will be a bit different, but still we'll get to the same place.
So there's this distinction between why are we doing all of this? Is it for drug development or is it for personalized medicines? This is going back to the first session yesterday. And you can sort of view this as if you think about drug development, we're looking at candidate genes. And if you're thinking about personalized medicine, we're hopefully thinking about something along the lines of polygenic scores. And so what we've seen in this research in the social sciences, typically they've moved away from candidate genes, very, very small effects, lots of concern, towards polygenic indices. And what I'm going to try to do today is talk about actually 3 research designs, although only 2 are in the slides, but I'll make some comments about a third. And the first step for either case is that you're going to start with this GWAS. And I'm going to also link this back to yesterday's talks on the GWAS. And I think where this talk is really going for, it may not really be for this audience who have a lot of expertise, but this is a lot thinking about new people who are coming into the field and they're just opening up a data set. Hey, look, there's a polygenic score. Let me toss it in. And then this is what I'm going to get, and this is what I'm going to conclude. And I'm going to be saying, hold on, you guys need to think a little bit more carefully. All right, so I'm going to be focusing a lot more on interpretation. I'm going to revisit 2 published papers by friends of mine, who may no longer be friends of mine after this, but we'll see. And I'm going to show you that what they state in their, well, you'll see. I'll hold you in suspense for the next 10 minutes. Okay, so we talk about GWAS a lot. This definition of hypothesis-free has been used quite often. And from Q's question at the end of Abdel's talk, it might be assumption-free, but unconfoundedness assumptions are everywhere in this thing. There's tons and tons of assumptions embedded in the specification of the GWAS. So essentially, we're running some polygenic trait on SNPs. SNPs we're going to treat as linear. Typically, we like to think about this as going from the minor allele to from the major allele, that'll be your 0, to the most minor allele, which could be 1 or 2. And then you have your pop strat controls. And then all your environmental stuff, and this is going to Abdel's big point, I think, at the end, is that it's all assumed to be sort of orthogonal to everything else in there, because it's all embedded in that error term, which I'm calling U. Now, what's sort of interesting is that as you start to look, and then what you include, which SNPs you include, all the linkage disequilibrium stuff that we've heard way too much is in there. Something which hasn't gotten a lot of talk about is that we're going to take these estimates from this and then to calculate our polygenic score, how we actually measure the SNP to calculate that polygenic score is not the same way that we included it in the regression. So typically, if you find a negative effect, the SNP is going to go from, instead of 0, 1 to 2, it's going to go to 0, -1, -2. This is called quality control. We can talk about that. And a lot of this paper is motivated from the idea about how easy would it be to replicate studies. All right, so why are we doing this stuff? Well, lots of these effects were really, really tiny. We've talked about this. One of the big assumptions that's going in this, and this is also related to Abdel's talk, is that you think about this because we have those pop strat controls. We're essentially assuming homogeneous effects on those alphas. So you're assuming that they're going to be basically constant across all time periods and across different populations, or where the different data sets that have been pooled have been. And what Abdel's talk was really about was that opportunities change over time. So should we really be having those coefficients being fixed? So that's going to be the base assumption that's in here. So there's a lot of assumptions in there. And I think where Abdel wants us to go is to basically say, let's start to relax. Is that about right? So, okay, good. Phew. Okay, so the first time I presented this paper, those of you who might have seen it before, was at UCLA. At UCLA, there was a famous paper written in the 1980s called Let's Take the Con Out of Econometrics. So I call this Let's Take the Con Out of Genomic Applications. And I think that there is a lot of excitement. I've been using genetic data since about 2002. I think it's a lot of fun. And the reason why I think it's fun is because it's, unobserved heterogeneity is deeply unsatisfying if we just toss in a fixed effect. This is sort of opening up that door to what is in that fixed effect. And the literature is very much in its early stages. And what I guess is troubling me and what motivated me to write this paper is there's a lot of evangelical claims I'd like to sort of push back against, and we can have a big debate. So we estimate our GWAS, that's the equation number 1. Then we calculate our polygenic score. And you'll notice that instead of having K's before the alphas, I have M's now. And that is because not all the SNPs that we estimated in that GWAS are in the data set where we're going to calculate the polygenic score. Okay? And because I really dislike this idea of changing the order of the SNPs because it's the quality control thing, I'm going to reframe this the second way with an absolute value sign. I think that's a much cleaner way of explaining how we calculate the polygenic score. Right? That way the SNP is the exact same order it is in the regression. It's just a personal preference. Okay. So now, when you read these papers, then you start to go crazy. And this was also in some of the questions that just came before. Which polygenic score are you actually using? Because there's a whole bunch that are created. And I think the transparency in these is often absent. And this is where the replication concerns come from. So which one do we pick, which one we don't? And I think the recipe matters, which one we chose. as well as the definition. So Delaney, Musselnova, and co-authors have this paper, which I wish I had written, and just basically says that which polygenic score you pick for a given trait could really change your results. And so she finds that the correlations between them go anywhere from 0.17 to 0.93 for cardiovascular disease, and from about 0.4 to 0.83 for educational attainment. So which one we're going to pick is important. Sometimes you could be in the bottom quartile with one, but then you're in the top quartile with another. Okay, so I'm going to go a bit further than what she's doing. I'm not going to compare different polygenic scores. I just want to look within a given polygenic score. Okay, so again, polygenic score, it could be the weighted sum, that's how I was defining it. It could also just be the raw count of alleles. Maybe you're only going to have maybe you're only going to sum the SNPs that are above a certain level of significance. There's lots and lots of different ways you can define a polygenic score. This is somewhat important. And the next point that I sort of want to make is that these GWASs, we're not really often, well, it's very, very rare we're just using something like OLS. We're typically using an estimator that is going to try to minimize mean squared error. So something like a ridge regression or a lasso regression, which might shrink a bunch of, and both of those are going to shrink coefficients. And so even for the educational attainment polygenic scores, these are not unbiased coefficients. They're coming from a Bayesian procedure, and they're a bit shrunken down. Okay, so I just want to make this point because we're going to go a bit further on that in a couple of minutes. There's lots of other issues that we could talk about GWAS, and a lot of them were talked about yesterday morning, so I'm not going to, that's for breaks. Okay, so Nick and Kevin are friends of mine. They have two great papers. One came out in Journal of Economic Association, the other with Dan Barth, who I don't know, came out in JPE. These are good at journals in economics, my discipline. They're both using the Health and Retirement Survey. They differ in which educational attainment GWAS is polygenic score they're using. One is using one from EA2 from Ockbay and Company, and the other one is from the Lee and Al EA3 paper. And the original question they were asking is, what does it mean when you're putting in these scores that associate with genetic endowments to explain a given other type of phenotype? And one, they're trying to explain earnings. So how does the educational attainment one explain earnings, and how does, or the other one's about family wealth. What I'm going to basically do is say, okay, when we're estimating these GWASs, we have about millions of SNPs. But a very, very small subset are significant. Remember that distinction between drug discovery versus personalized medicine. So what I want to do is I just want to decompose that into the significant part and to the insignificant part. And this may change the story. And this is going to be very important for how do we interpret the results. Especially if we want to convince policymakers that this stuff matters, we better be hoping it's coming from the right part of the polygenic score itself. So the plan is very, very simple in this paper. I'm just going to decompose the polygenic score into two different pieces. I'm going to take it as constructed in the HRS and then just say this is from the significant genes and this is from the insignificant genes. Why am I doing this? Well, you read the abstracts of the paper. The first one from Barth and Al, they say they're providing evidence that genetic endowments related to human capital accumulation are associated with wealth, Papa George and Tom are basically saying that their evidence is providing, they're showing patterns that suggest that genetic traits that promote education might also allow for . So reading this, what we're hoping for is, this is really coming from the significant part of the polygenic score, not from the insignificant part. And now you guys can take bets. Okay? Now there's lots of ways to calculate polygenic scores. There's lots of softwares out there. There's even new softwares we learned about today. Great. The nice thing about these softwares, they're publicly available. What's the key thing? So let me just jump down to, and then I'm not even getting into the imputed SNPs, which is another issue that we heard about that yesterday morning, but we can talk about that more if we have time. So in educational attainment 3, about 5,000 SNPs are significant, and about 1.5 million are insignificant. So when we're going to be using this data from the HRS, they only have 1,050 of the 1,271 that Lee and Company say are significant in the data, and those are going to be the ones with the coefficients from Lee and Al that we're going to say this is the significant part of the polygenic score, and the residual stuff, the difference between the HRS polygenic score less that, is coming from about 8 million genes. That's what we're going to call the insignificant part of the polygenic score. And then we're going to do the same thing for Ockbay. We have 64 to 74 genes and 7.5 million of the insignificant ones. Okay, this is just a heat map. It is totally unreadable, but the whole point of this heat map is to basically say on one axis is where you are in terms of the size of your significant component of the polygenic score and where you are on the insignificant part of the polygenic score. So if these things are, this would not be a concern if everything was right around the main diagonal. But because sometimes people have very low significant scores, but very high insignificant scores, this is why this decomposition might be important. All right, so let's get to the results. We're going to start by replicating table 4 of Barth and Al, and we're getting it. So basically the top part of the table is theirs. That's their results. And what you can see is, look, there's where they reached that conclusion. In columns 1, 2, and 3, you can see that highly significant polygenic score coming in. In part 2, down below, we're basically breaking this into the two components. And what you can see is in column 2 and 3, yes, the significant part is coming in, but the effect is much smaller than the insignificant part, which is in column 3. Which is here. And we can actually test, are these effects offsetting? Are they equal? We can reject that they're equal. The main effect is actually coming from the insignificant part of the genes. So this is actually changing the conclusion, and in several of the specifications, all the effect is coming from the insignificant part of the genes. Let's go to the other paper. So that's their result at the top part, and then we're going to break it down into two pieces, into the significant and insignificant. Same type of story, the effects that they're finding about how this gene is related to educational levels, and they're going to consider different sheepskin effects, that's language from econ, and we're going to find that most of that effect is all coming from the insignificant genes, not from the significant genes. Where the significant genes actually come in, or a significant component of the polygenic score comes in, the effect is much smaller in size, and again, at the bottom of the table, these effects are not offsetting. Okay, so this is the general evidence, but now let's head to a little bit more things thinking about applications of polygenic scores. One thing which we didn't talk about that much at this conference is, but it did come up, instrumental variables, sometimes called Mendelian randomization, although it really should be called Mendelian encouragement. The basic idea there is that you're going to use that polygenic score for an instrument for some type of trait. So Jason and I have written papers where we've actually done gene environments in another lifetime. Jason's pretending he doesn't know me anymore, but that's okay. And in those, we were using candidate genes, and of course we got criticized. How do we know that these candidate genes really only affect the outcome through this endogenous regressor? And of course, we hand-waved and we did our best. We did over-identification tests. No one believes us. But if you're going to use a polygenic score for an instrument, people say, well, I can't do an over-identification test because I'm just identified. I have one endogenous regressor, one instrument. But if you think that having like 4, do we have 4 genes, Jason? 4 candidate genes? About that.
4 to 6.
Maybe 6, I can't remember. Jason did all the hard work. I just tell the stories. If you think that 6 genes could violate could be a concern here, you're using about 8 million of them. So that's kind of crazy, right? So this is sort of nuts. But that's comment 1. The other comment is that if you really think about what the polygenic score is, now this is in economics, we have this things called Bartik instruments, which are now called shift share instruments. This is a big popular research design where you're using a certain type of instrument, which has 2 pieces. It has an effect that's estimated, and then the shift, and that's the share. And there's a whole big debate about what is really driving the effects. Is it the shift or the share? This is what the polygenic score is. It's a shift share instrument. And what's really driving it here is these shifts. And so that means that you really need to have exogenous shares for this to work. That's a very, very strong assumption. But I was thinking quite a bit about this shift share instrument, and this is about another research design. So if we're going to think about using the polygenic score and talking about in a regression and talking about having a causal effect, it doesn't matter how many levels of ancestry you put into your equation. To have an interpretation, you have to fix either the shift or the share. So which one do we want to fix first? The shift or the share? Let's fix the shift. So let's say I fix the shift. That basically, if I fix the shift, and my interpretation is all coming from changes in the share. That's like CRISPR type design. And that basically means that, going back to Abdel's earlier point, you can't have population changes or time-dependent changes in those shares. They have to be fixed parameters over time. The other way around, if you're going to have basically the shares changing, so you're going to be identifying off shifts, that's basically assuming that all mating has to be random for you to use this. So if you're going to be using this, you have to really think carefully about what are those assumptions to interpret this properly. So, okay. Next point is the other strategy. This goes to the session. It's about, let's throw this in as a control, and let's look at gene-environment interactions. Okay, so first of all, number 1 is that you should be estimating the GWAS jointly with this equation, and then bootstrap your standard errors, because otherwise you're using a generated regressor. That's one point. And the other point that I want to sort of make about this is that you have to be really, really careful about interpreting this. And there's a lot of so I think estimation error in the score itself is much more important than issues like measurement error. And the reason is that all the discussion about measurement error is thinking about this as random measurement error, classical measurement error. But really, if the estimator that we're using to do the GWAS is shrinking estimates, all those coefficients are biased downwards. Now your coefficient's biased upwards. So this is actually a different type of concern. So I think you have to really think much more carefully about the estimator you're using. Okay, my last minute. So what I sort of argue is that estimating the reduced form, doing something along the lines of a candidate gene type study with lots and lots of candidate genes, and then shrinking that set, doing like an OLS post-lasso, is probably a safer way to go. Okay, That's what I think we're at. So I think what this paper is really trying to about is about trying to think about how that polygenic score is created. You really need to think much more carefully about it in order to interpret its effects. And I think interpreting the effects is going to be, and interpreting them correctly is going to be very, very important for this field to move forward in the coming years. This audience is clearly big supporters. Let's get social genomics to be much larger so we can have bigger and better conferences and a whole bunch of things. And I think that the credibility of this is going to depend a lot about the interpretation. So I think we need to think about this a little bit more. Okay, so that's what I say there. So the other point that I kind of want to make is that polygenic scores, and there's lots of these software packages, the dangers of automated sophistication, just plugging in a line and then using it. There's lots of assumptions embedded. We have to understand what they are. And I don't think that they're trivial or they're minor. We have to think very, very carefully about it. I think we have to make it very, very clear what assumptions we're making in our analysis. All types of work requires assumptions. Let's just be explicit. I think that for a lot of people, jargon remains a very big barrier to entry. And you guys are probably saying, yeah, listen to Mr. Econometrics jargon up there. But jargon, I think we have to sort of open up the door and sort of shine more light so it's a lot clearer. I will say that there's a lot of concern that we really don't understand that much about genetics and exactly how they work, but I don't think that should be a real big concern. Macroeconomists have been studying macro phenomena for years. They haven't gotten anywhere. So this is all okay. Like you know, progress moves slowly. And I really think that for policy purposes and for this research to get much more attention, society needs to develop a much more healthier relationship with our genes. And I think that we're sort of go from like super fans on one side or into the eugenics and exceptionally phobic. And I just think, you know, being a bit more honest about the pros and cons and interpreting things carefully is the key forward, so we don't end up in more tribes. We have enough other tribes elsewhere. I'll stop there. Now you can attack me because I was deliberately provocative. I'm an economist, so you can be as harsh as you want. I'm used to it.
Actually, I'd like to thank you for your voice of skepticism. This is a fascinating field. I'm a layman at best, but I think perhaps, and you point out, we may be rushing too quickly into conclusions and theories as opposed to doing more strict research moving forward. So I'm not attacking, I'm thanking you for your observations.
Okay. This has never happened to me before, so okay. I should have recorded. Oh, it's being recorded. Okay. My wife wouldn't believe it otherwise. Okay. Phew, yeah, this should be fun. Okay, let's go. Gloves are on.
No, I actually completely agree that we need to be careful when interpreting, these parameters we estimated through a polygenic score-based approach. I just want to comment on the fact that I think you produced the two different papers' main results and split the polygenic score into the significant versus non-significant one. That is completely expected. I think in complex trait genetics, people know that it's just because the top so-called significant findings. I think you showed the list of SNPs you included in the score. It's a very small fraction of the genome. Their effect sizes are not much larger than the remaining of the genome. If you split trait heritability based on whether SNPs are significant or insignificant in the GWAS, you will see exact same pattern. So that is something people have realized maybe 15 years ago. But when you use regularization to produce scores with biased SNP effects, can you still interpret effect sizes obtained downstream using these scores? I think that's a fair point. We need more methods to address this type of issue. I think you made a minor point about whether or not you can add this very noisy score to control for genetics, to control for genetic confounding. In the next section, you'll see a talk to address this without actually calculating polygenic scores directly from GWAS statistics. Maybe you can be happier with that type of method development.
This means you can't go home. Okay. Okay, so now everyone's excited for the next session.
We have one more.
Oh, we have another question. Excellent. You're getting a lot of exercise from one corner to the other. So, okay.
So that was quite fun, actually, I think, right? Did you have fun? I enjoyed listening. It seemed like you were having fun.
It was more garbling at the beginning. But,
no, it was good. But I wonder, in the end, are you promoting a particular approach to the computation of polygenic indices? Because you said something about okay, we can do something like lasso and we'll downweight some and upweight others. Is that what you're promoting? Like
no.
Where do you land on how to do the
So I kinda
computation of
like-
PGIs.
So I kind of like the idea of paper 2 yesterday. Is that your Chen? Is that who gave it? I kind of think that we have to start to think about, let's have gene by gene interactions. So let's say, the model, well, first of all, why are we going 0, 1, 2? We could just dummy the things out, keep the major allele out. That way we're not enforcing linearity on everything. So we could just be much, much more flexible, and then we can even allow for, what I like to call, but nobody ever picked up on this term, environmental stratification. To pick up general environmental differences, throw it in there, and then we're getting sort of step 1 towards incorporating those things that were previously incorporated. So I think the GWAS that we're taking is, we're stuck in this old style of GWAS. And I think this is the second session where I'm basically saying, maybe step 1 is let's think a bit more carefully about how we're doing that GWAS. And then if we're interested in drug discovery, then we care about unbiased coefficients. But if we just care about polygenic scores, let's do some machine learning stuff, let's minimize mean squared error. But then we have to be very, very honest in the next step that this is being generated from biased coefficients. But what is the goal? And I guess what I kind of view this is that, and I agree with Q that a lot of these results aren't new, but it doesn't stop from people from making big claims when they're writing papers. So I just sort of want to, highlight things that yeah. Go ahead, Sol. Sorry.
Thank you, that was fascinating. I mean, you made some bombastic claims, but I think some are sort of misguided. The last one that you said about linearity, the last OCPA paper, they relaxed that, and they realized that it didn't make any difference. And anyone that has worked with this polygenic scores, you take like the EA, and using, I mean, as an applied person, I also have the situation, like, you're estimating this in one context, and then you're applying a different one. But anyone that sort of like has taken the EA PGI and sort of tried to see across different samples and even estimating causal effects using the family designs, it's sort of remarkable how stable and sort of like comparable those results are. Much more than like when you look at sort of effects of environment and you try to, in different data sets, it's just sort of remarkable how. So I don't think anyone here is denying that there's room for improvement. But yeah, I think some of the bombastic claims are just sort of misguided.
But I think we could do it it would be very easy to just do an F-test. Can we restrict our linearity, right? So at the end of the day, because of, if you think, if we have misspecification and we're having, and the model is overfit, you're not gonna, of course, the end of the day, PGI is gonna be very, very similar, the total number. That's what's going on there. But the big question that I think that the easier way to test that would just be to do F-tests on each and every single one, which is essentially sort of going back to Wei Min's talk, which is you're fine-tuning, you could see actually where it actually holds and where it doesn't hold. That would actually be interesting to me. But, yeah.
All right, thank you. We should move on to the next session. You got what you wanted.
I want to wake it up.
Yeah, you did great.
We don't have a break in between, right?
No.
Well, this actually perfectly sets up the next session because the topic is going to be polygenic score applications and methods. Let's welcome our panel speakers and join us in front. Okay, so polygenic scores are obviously a very important tool and a topic of great interest in human genetics and social genomics. And it's our observation that in the general complex trait genetics community, a lot of people care about identifying people with particularly high genetic risk for certain trait, and then think about early intervention and prevention and diagnostic screening, that kind of thing. In social genomics, this seems to be less of a concern. The use case, based on our observation of polygenic scoring the social genomics context, seems to be twofold. First type of application is to do some kind of gene-environment interaction. You want some kind of a proxy for the G in G/E studies, and a lot of people are using polygenic scores as this dimension reduction kind of approach instead of using millions of SNPs directly in the model. And another type of application is to add polygenic score as some kind of a covariate in a sophisticated regression. People are fundamentally interested in phenotypic relationships between some kind of exposure and some kind of outcome, but then people want to control for genetics. And how do you control for genetics instead of adding millions of SNPs into regression? Let's add a score into the model. Hopefully that is sufficient to control genetics. And I think in this panel, we're going to see, well, we have seen a lot of discussions on G by E already, but in this panel, we're going to see some approaches to tackle this problem about how do you correctly remove genetic confounding in social genomics related topics. And from a methodological standpoint, more and more people are beginning to realize that there's an urgent need to generalize our human genetic analysis to diverse populations. When you do that, you realize these polygenic scores were mostly developed based on non-Hispanic white populations. When you port these scores to diverse populations, they have terrible, terrible results. So it's a very hot topic in method world. How do you develop improved approaches to increase the portability of these scores, which is also something we're going to see a little bit in this session. That's my overall intro to the session. And let's welcome our first speaker, Dr. Robel Alemu from UCLA and Broad. He's going to talk about relative predictive performance of PGIs across diverse ancestral populations. The stage is yours.
Thank you. Hi, everyone. Good morning. My name is Robel Alemu. So I'll be presenting a project that looks at the relative predictive accuracy of polygenic indices across genetically inferred ancestry. So this work is in collaboration with Patrick Turley, Aisu Okbey, and Dan Benjamin. So I think most of us have heard a lot about genome-wide association studies. So over the past decade or so, the sample size of genome-wide association studies has been increasing very rapidly. And that has led to the identification of genetic variants, specifically SNPs, that are replicably associated with a wide range of traits and disease conditions. And this increase, however, is very unbalanced in a way because most GWASs have been conducted on one genetic ancestry, specifically European genetic ancestry, while other non-European genetic ancestry are severely underrepresented. There are some improvements, as you can see in this chart, in recent years, mostly to do with increases in the representation of East Asian ancestry, but other non-European ancestry are still severely underrepresented, especially if you think of the global population and what these genetic ancestries represent in terms of the percentages, then you see clearly there is a huge imbalance. So, different studies have shown that with the rise in the sample size of genome-wide association studies, the predictive accuracy of PGIs, which aggregate association information across thousands of SNPs, have increased in terms of their predictive accuracy. But this is unbalanced across genetic ancestry, given that most of the discovery sample is based on European genetic ancestry. So these increases in the prediction power of PGIs are limited to European genetic ancestry. And a seminal work by Martin et al. 2019 has shown that relative to a reference group, which is European ancestry in this case, by looking at mostly biologically proximal traits, around 17 biologically proximal traits, they have found that the relative predictive accuracy of PGIs declines substantially as a function of genetic distance. So genetic distance relative to the sample that was used to discover the associations, basically. So relative to European genetic ancestry, they find that the predictive accuracy of PGIs on average declines by about 80% in African ancestry, about 50% in East Asian ancestry, and about 40% in South Asian ancestry. And this is huge. And it has huge implications because the poor transferability of PGIs across genetic ancestries would prevent the equitable potential use of genetic analysis across genetic ancestries. And this may exacerbate existing disparities. So a bunch of factors have been implicated with the reduction in the prediction power of PGIs.
And the most common ones are cross-population differences in allelic frequency and linkage disequilibrium between the causal SNPs, which have been indicated in earlier presentation that we don't know mostly of, and the SNPs that are tagged in genome-wide association studies, which we call PGI SNPs. And other factors, including differences in heritability, gene by environment interaction, and other factors have also been shown to play a big part in terms of the reduction in the prediction power of PGIs. A very seminal work also recently by Wang et al. 2020 derived a theoretical model to get at this issue of understanding what really explains the drop in the prediction power of these PGIs across genetic ancestries. And they defined the relative accuracy as a function of these really important population parameters, which I will be talking about more of. But this study has only looked at very few biologically proximal traits. And not a lot has been done on traits that we all care about. So more complex behavioral, social, aging-related health conditions have not been studied well. So in their study, Wang et al. Has shown that the loss of the PGI prediction accuracy that's explained by two important factors, which are minor allele frequency differences and LD differences, account to a really large, over 85% in African genetic ancestry and nearly 60% in East Asian and 40% in South Asian genetic ancestry. So what we, in this study, aim to do is sort of expand the scope of the phenotypes that we're looking into, not just biologically proximal traits, but a wide range of traits, but also tweak on how we really go about estimating the factors that are contributing to the loss in the prediction power of the PGIs. And this is what you see here. So we're basically relying on the UKB and sort of validate our findings looking at the HRS. And we'll be looking at a really long list of phenotypes. In terms of the methods, we employ a bunch of methods. First is identifying the genetic ancestry of our sample. So we do that by using the PCA-based approach that's suggested by Yang et al. And to compute the PGIs, we use a Bayesian-based approach, which is S-Bayes-R. So the Wang et al. Paper that I had mentioned earlier used genome-wide significant SNPs, and they use pruning and thresholding to compute the PGI. So that's an improvement in this regard because we're using genome-wide SNPs to compute the PGIs, which are shown to be more predictive. And we compute two types of relative accuracy estimates. One is the observed relative accuracy, which I'll talk about how we calculate it later on. And the other one is the expected. So the expected relative accuracy is based on the theoretical model that tries to estimate relative accuracy as a function of population parameters, which I'll be talking about more on. So we implement a slight modification of the Wang et al. Approach and account or explain the loss in the prediction power of PGIs as a function of LD, MAF, and SNP-based heritability. So here's the model that I'm talking about. So we sort of decompose the relative accuracy. So what do we expect the relative accuracy to be if we model it based on these important population parameters? And you can see here, this includes the squared correlation of causal SNPs between our reference group, which is European ancestry, and our target groups, which are non-European ancestry. We set these to 1, we assume, so make that assumption. So, but that's in reality, that's not the case, but we make this simplifying assumption. And also, the next aspect is basically the ratio of the SNP-based heritability between the reference and the target population. And we also have LD correlation between the causal SNP and the PGI SNP. So mind you here, we don't really know the true causal SNPs of different traits, So what we do is we follow like an iterative approach. So we identify top SNPs, 3 sets of top SNPs that are associated with traits, top 100, top 1,000, and top 10,000 SNPs that are associated with each trait. And then we also identify the SNPs that are associated with these top, that are in LD with these top SNPs. And we consider these SNPs as quasi-causal SNPs. And that's how we also advance relative to the Wang et al. Paper, which only based the results on genome-wide significant SNPs, which are really few, especially if you want to look at really complex traits which are influenced by thousands of SNPs. Just looking at genome-wide significant SNPs won't get you far. So after that, what we do is basically to compute the observed relative accuracy, we compute the ratio of the incremental R-square between the target and the reference group, and we also get the expected relative accuracy, and we compute the loss of accuracy due to these important population parameters as 1 minus the relative accuracy. And then eventually what we do is to get to the share of the loss in the prediction power of the PGIs that's attributable to these factors, we take the ratio of the loss of accuracy. So the expected loss of accuracy over the observed loss of accuracy due to these factors. So a couple of assumptions. We assume that causal SNPs are shared across ancestries, and the effect size of these causal SNPs are perfectly correlated across ancestries. Of course, this is not true. This is a simplifying assumption. And another limitation is that we only study common SNPs, so for computing the PGI. So this may not generalize to rare SNPs, which often are population specific. And we also have this limitation of, because we are inferring, so in the expected predictive accuracy, we infer these important population parameters from reference panels, the 1,000 Genomes Project reference panel. But these may not apply, translate in admixed ancestry. So some results. So if you average across all 43 phenotypes, you clearly see that the observed relative accuracy is much lower in African ancestry relative to the other ancestries, followed by East Asian and South Asian ancestry. And this, relative to the last study, which is Martin et al., we clearly see that there is substantial deterioration in the relative predictive power of PGIs when you include much more complex traits. But it's very much similar for African genetic ancestry, which is about 20%. So if you disaggregate these into phenotype categories, you clearly see that the observed relative accuracy varies by phenotype category, and the highest relative accuracy is in anthropometric and cardiometabolic traits, as expected, and the South Asian genetic ancestry has the highest observed relative accuracy across the board. So the most interesting result that you can see here is that the contribution of minor allele frequency and LD to the loss in the prediction power of the PGIs across phenotype categories, you can really see that the share of these two factors for African genetic ancestry especially accounts from 70 to 100%. So the loss of this prediction power in African genetic ancestry is largely due to these factors, whereas in East Asian and South Asian ancestry, with exception of health indicators where you have like some differences and cardiometabolic traits, it's largely, it's very small. The contribution of these two factors is relatively small. So if you go deeper into looking at what happened in cardiometabolic and health indicators, you clearly see some interesting results. So one is that, for instance, for coronary artery disease, you see the contribution of these factors in East Asian and South Asian ancestries is really low. Whereas in traits such as LDL cholesterol, so the same pattern, but for the other cardiometabolic traits, the contribution of these factors for East Asian and South Asian ancestry is relatively large. And the results that we find for LDL is actually quite different from what Wang et al. Found. So based on their analysis of just looking at genome-wide significant SNPs, they find that the contribution of these two factors for LDL for these ancestries is really large. And that's a stark contrast to what we find here. And there are some interesting patterns as well, as you can see for health indicators such as for pollen allergy, for migraine, and for hay fever. You clearly see a deviation in a way that the contribution of these two factors is relatively larger compared to other phenotypes in the category. So we also do the same analysis in another cohort, which is the HRS, and try to map this out, the contribution of these two factors in UKB and HRS. But this is strictly for African genetic ancestry because we don't have enough sample for other ancestries in the HRS. So we clearly see substantial differences in the contribution of these two factors in these two contexts, different contexts. So African ancestry, but living in different parts of the world. You see the contribution of LD and MAF, for instance, in UKB for certain traits such as drinks per week and subjective well-being are much significantly larger. And conversely, you see also some other traits where the contribution of these two factors are much higher in HRS relative to the UKB. So this clearly shows you like the contribution of these two factors is one, context dependent, and is also phenotype dependent. So that's the key result here. And what happens when you account also for SNP-based heritability? So the two factors, as I indicated, explain different segments depending on the phenotype in the cohort. But what happens when you account also for SNP-based heritability? And this we were only able to do in the African genetic ancestry because the SNP-based heritabilities were imprecisely estimated for other ancestries. And you clearly see that once you account for SNP-based heritability, for most traits, the loss in the prediction power of PGIs gets to almost 100%. So you get to explain the loss in the prediction power by just accounting for differences in these three major population parameters. And the same happens, the panel in the right is showing the results for HRS, but for just few traits. And you clearly see that. So just to conclude, we find that the shrinkage in relative accuracy of PGIs in African genetic ancestry is mostly due to MAF and LD, and that the contribution of MAF and LD is much smaller in East Asian and South Asian ancestry, and there are notable differences across phenotypes, and the lowest contribution we see in cognitive, personality, and psychiatric traits. Other factors such as gene-environment correlation may explain the rest of the loss in the relative predictive accuracy of PGIs, especially in East Asian and South Asian ancestry. And our results are consistent with what Wang et al. Finds, with few exceptions, especially LDL, cholesterol. We advance literature, especially Wang et al., in a couple of ways. One is expanding the scope in terms of the trait and the context, and also using more predictive PGIs instead of pruning and thresholding using SPSR, and also improving on how we go about estimating the relative predictive accuracy. And we detected differences in the contribution of these two factors, MAF and LD, in African genetic ancestry, populations that are residing in different parts of the world. That's UK versus the US. And eventually, all these results sort of underscores that the varied impacts of these parameters depends on the trait that you're looking at and the context, which is the environment that populations live in. So with this, I finish my presentation. I would like to thank SSGAC, UCLA, and the Broad, Institute of MIT and Harvard. I'll be happy to take questions. Thanks so much.
We have time for maybe one quick question.
So in my experience, the African ancestry generally have more variants in the LD region. So is that what explains the large difference in LD and MAF?
What region?
So in my experience, the African ancestry have the most variants compared to the other ancestry. So is that contributing to the dropping of the RA?
Yes, so that's part of the story. So the thing is like, so if you think of, so this is relative to a reference group, which is European ancestry. So you clearly see substantial differences in LD and MAF with the reference group. And that happens to explain the huge drop in the, because the training was based off of like the reference group. So if we had like used an African genetic ancestry as a training set, then the story would be different. But given that we're using European genetic ancestry, where the genetic distance between European and African genetic ancestry is really large, our study is showing that these factors are actually responsible for that drop in the prediction power.
Another question.
So we have to move on, but because of time, but of course we can follow up with the speakers after. Yeah, our next speaker is our almost Dr. Zijie Zhao. He's a PhD student in our biomedical data science program here at UW-Madison. He's going to defend his dissertation in just a couple months. And he's going to talk about controlling for polygenic genetic confounding epidemiologic association studies.
So thank you for the introduction, Q. So hi everyone, welcome. My name is Z, and I'm a PhD student in biostat department here at UW-Madison. I'm trained in statistical genetics, and I work with Q on the methodology and applications of polygenic score. So it is my pleasure today to talk about our recent work about controlling for genetic confounding in observational data analysis. And this is a collaborative work between fellow lab members, Dr. Sylvia Barcelos and Dr. Jason Fletcher. So many social scientists epidemiologists are interested in identifying social and environmental risk factors for health outcome. For example, many people in this room would be very interested in studying the effect of an additional year of education on various later life outcomes, including cognition, overall well-being, or longevity. It is well established that to do any sort of valid inference on association study, it is crucial to control for any confounding factors that contribute to both the exposure and outcome variable. And then there are some study designs that are specifically designed to address this kind of confounding. For example, twin and sibling designs leverage the shared family and genotype similarity between twins and siblings to control for confounding. Natural experiments such as the educational reform in UK in the 70s mimics randomized control trial, and it is usually free from any confounding effects. As you can see, there are these are all examples of studies trying to investigate the effect of EA on various health outcomes through these various study designs. And the issue with this kind of study design is that they typically come with small sample size, and also they are not always ready for researchers to use to investigate, to do this kind of association analysis. And typically, the kind of data set that we have is this large population level biobank. So these are very powerful designs with large sample size, and they are generally available to many researchers. And the issue with the downside of utilizing this kind of large data set is that now you really have to control for the abundancy of potential confounders. So one source of confounding effects come from genetics. So now we know that many human traits have a substantial genetic basis, are extremely polygenic, and when you look at any pair of phenotypes, they may share a substantial genetic basis. So this pervasive pleiotropic genetic effects from single nucleotide polymorphism or SNPs onto these complex human traits can confound the association analysis on the phenotypic level. So for instance, when we investigate the effect of EA on later life cognition, we know that these two traits share substantial genetic basis. So it's really important to account for that pleiotropic effect in association analysis. So like any confounders, genetic confounding effects can lead to spurious association when it's not accounted for. So here is a rather kind of circular evidence that I can find that's related to medical aspect of this kind of association study. So for example, here on the bottom left, this is a screenshot from American Diabetic Association titled "Low Vitamin D May Contribute to Insulin Resistance." The evidence of these claims come from association findings between low levels of vitamin D and increased risk for type 2 diabetes. So on the top right corner, this is a meta-analysis of clinical trials saying that there is no found evidence to say there is an effect of vitamin D3 supplementation on diabetes prevention. And then lastly, on the bottom right corner, this is a recent GWAS paper for vitamin D. And then in that paper, they concluded vitamin D level and type 2 diabetes to have significant and negative genetic correlation. So putting everything together, there can be multiple interpretations as to what's really happening here. For example, it may be a reverse causation issue where getting a type 2 diabetes actually leads to lower vitamin D. This explains why you see a phenotypic association, but you don't see any causal effect. Another hypothesis may be that there may not be any effect from vitamin D level to type 2 diabetes. And the reason you are seeing a phenotypic association but not causal effect is because this is because of confounding factors, including this genetics. Because if there's no genetic confounding at all, you wouldn't expect to see such substantial genetic correlation. So people certainly notice this issue, and there has been a lot of previous studies trying to control for this genetic confounding in association analysis. The common practice is to control for PGS, and this is a very common approach. Here are two examples. Basically, the first example investigate the effect of loneliness on depressive symptom and also adjust for the PGS of loneliness. The second example adjusts for both the PGS of the Y and the X. So as you can see, there are no consensus over which PGS to control for in this kind of association analysis. And the conclusion from these studies, and also some that I didn't list it here, are kind of consistent, saying that adding PGS in the association study didn't really change the effect size or the significance between the variables that you are interested in. So we start to think about the issue of using PGS to control for genetic confounding. So the model that we really want to fit is on the top right corner. This is, we want to actually add the true unknown genetic confounding effect between Y and X in this regression. But empirically, we couldn't get, we don't know what that G is. So what researchers can do is to replace this G by an empirical version, which is the PGS. Now, the use of PGS will give multiple issues. First, we have to ask which PGS do we control for. There is no consensus over which PGS to control for, and this is entirely hypothesis-driven. And this kind of hypothesis is very difficult to validate in practice. Second is PGS is a very noisy estimator, and it has huge measurement. It can have huge measurement error. So adding PGS in the regression model may not be sufficient for confounding control. And then thirdly, from a practical standpoint, calculating PGS requires independent GWAS at testing data, and then this kind of data requirement may be too stringent sometimes, and we may not have this kind of data in practice. So a better way would be to control for just all the SNPs that we have, because we know that these genetic confounding effects come from these SNPs. And then if we can control for all the SNPs in the data, then we can control for all the genetic confounding. And here is the model for that. So basically, we have an exposure X and outcome Y, and X has an effect on Y, and then two traits are both heritable. So the G here is, instead of the genetic confounding effect, is actually a vector of genetic variance. So they all have this genetic basis. So in order to get an estimate for this B, what we can do is to regress all the SNPs from X first, regress all the SNPs from Y, and then we can do this regression between residualized X and Y. And then in the part highlighted in blue, we can see that even though we cannot fit this kind of linear regression in practice because of the huge dimension of the genotype data, with unlimited sample size, this is how the point estimate for B will look like. So it's basically a difference in the difference between phenotypic covariance and genetic covariance over the difference of phenotypic variance and genetic variance of the exposure X. So in this way, we can see that we can just plug in estimators for each of these four terms, and then we can get this estimator for the effect size B. So basically, everything here on this part, the bottom part, can be estimated based on either testing data or GWAS summary statistics using software such as LD score regression. And then in this way, we have a point estimate for this B hat. And I omitted some details here. We also derived the sampling error and also developed formal statistical tests to directly test the significance of this exposure effect. And then one last point in this slide is that this is an interesting observation because when we use LD score regression to do any genetic correlation, genetic covariance analysis, we know that the intercept term of this LD score regression will actually capture the phenotypic correlation if the two GWAS summary stat has overlapping samples. So this really means that we can replace the phenotypic covariance estimation based on the intercept, based on transformation of the intercept from LD score regression. So in this way, we can do everything based on GWAS summary statistics without actually accessing any individual level data. So to summarize, we formally introduced Penguin, polygenic genetic confounding inference for effective control of genetic confounding in association testing in analyzing large biobanks. And then the key intuition of our approach is that the gold standard regression we want to fit is to control for this unknown true genetic confounding effect. And this is equivalent to just controlling for all the SNPs in the data. And then comparing to the PGS approach, our approach is hypothesis-free in terms of the underlying nature of the true genetic confounding effect. And we, in addition to Penguin, we also introduced a full summary stat-based version, like I described earlier, called Penguin S. So first, we evaluate the statistical property of our estimator in simulation. So we conducted simulation in WTCC data. We simulated simulation settings, various simulation settings with various confounding degree and different exposure effect. And then we replicate this simulation 100 times, and then we just look at the distribution of effect size from different methods. So if we look at panel A, from left to right, these box plots really are just our methods, Penguin and Penguin S, and then G-sense X and G-sense Y, which are structural equation model that uses PGS of X and Y as input, and then directly control for PGS of X or Y in the regression model. And then the last box plot would be just marginal regression between X and Y without any genetic control at all. So as you can see in this kind of model in panel A, where the x-axis is the estimated effect size, y-axis is estimated effect size, x-axis is just different settings with the true effect denoted as the red dotted line. We can see that Penguin and PGS and G-sense X actually give you unbiased estimate. And then using PGS to control for genetic confounding will give you highly biased estimate. And then in panel C, this is just to show the coverage of different estimator, and our method has the best coverage, 95% confidence interval coverage among all settings. Then we also introduced the sensitivity analysis in panel B and D, just to introduce some sample overlap between the testing data and the GWAS training data for PGS construction. And we can see that in this setting, our method is the only one that can handle sample overlap between data sets. So we moved to real data in UK Biobank, and then we chose to study EA because EA is an important indicator for a lot of later life outcomes, and also it is modifiable. It has a substantial genetic basis. It shares substantial genetic basis with lots of phenotypes. So that's the reason why we chose EA. Then we curated a list of 25 phenotypes from these two papers, and then we compare our methods versus controlling for PGS of EA and versus not doing any control at all, which is marginal regression. So in this plot for panel A and B, on the x-axis, this is the estimated EA effect based on different methods, and each line is one trait. And we can see that overall, when we look at all these traits, the reduction of effect size from our approach, which is Penguin, comparing to marginal regression, is much larger to the median reduction in effect size comparing PGS control to marginal regression. And then in panel C, this is just to show that the summary stat-based version of our approach and and the usual approach is really consistent in terms of the effect size estimation. So the full summary stat approach has great potential in real data analysis. Then lastly, we are also aware that many researchers are interested in identifying the effect of parental outcomes on children's developmental outcomes. And then here, genetic confounding is really something to be controlled for because of this shared genetics due to Mendelian genetic transmission. So we extended Penguin's framework into this two-generational model. So basically, the things that are changing is basically the things, all the terms on the denominator. So first, we need a phenotypic covariance between, using a maternal phenotype as an example, the phenotypic covariance between maternal and offspring phenotype, and also the genetic covariance between offspring and maternal phenotype. And the genetic covariance can be estimated using one GWAS summary stat by regressing offspring phenotype on the maternal genotype and another GWAS summary stat by regressing maternal phenotype on maternal genotype. And then again, we did this analysis in health and retirement study where we have each participant's own maternal and paternal years of education. And then we compare Penguin with using PGS as control and marginal regression. Here in the plot, the y-axis is the estimated effect size. Different colors means different method. Our method is highlighted in blue, and this really shows that using this method will give you a larger attenuation in effect size. So to summarize, we developed a reference component a variance component estimation framework to effectively control for genetic confounding in large-scale observational data analysis. This is hypothesis-free in terms of the underlying nature of the confounding effect, and also it can incorporate flexible data inputs. And we also show that, which is very important conclusion, is that adjusting for PGS in this kind of regression analysis is not sufficient. And there is substantial genetic confounding in real data when we do this kind of association analysis. And then our preprint is shown here. So if you are interested in seeing any more details, feel free to check it out. We also have a published software in GitHub. So everything is compiled into this very nice R function. So feel free to try this out and let us know what you think. So lastly, I want to thank all the co-authors, my advisor, and for their contribution and help for this project. And I'm happy to take any questions.
Thank you. This was great. So if I understood correctly, you said that when you use Penguin, you also have your coverage is better, so like you have tighter standard errors. Is that right?
Actually, it's the inverse. So the standard error will actually be wider because we have to take into account all the sampling variance of the estimators that we plug in. And then usually the estimator from LD square regression will give you slightly larger confidence interval comparing to estimator you get from individual level data. But it is because of that, because we take into account all the sampling variance of the estimator and their covariances so that we end up with a wider confidence interval. And it turns out to have better coverage than any other method because those methods have smaller confidence interval, and that leads to bad coverage and biased results.
I see. But is it a correct understanding that, say, there is kind of a trade-off there between bias and variance? Because, like, you guys are not biased, but then you have greater variance.
I guess it is fair to say that, because if you look at the variance of all this PGS method, the variance will be very small, but it's highly biased.
Okay. Thank you. That was great.
Thank you.
Quick question, because I'm a bit confused. So when you're looking at all the outcomes, can you allow them to be correlated, or all the errors in those have to be separated also?
By errors, what do you mean?
So when you were showing the effect of education on 15 different outcomes.
Yes.
Are you assuming the errors in each of those are independent of one another? Or are you sort of estimating them in a group, like a seemingly unrelated regression?
So in each analysis?
Each analysis is separate.
It's
are
separate.
They independent or are you allowing for correlations is what I'm asking.
So for each trait is a separate analysis.
Okay, so can you group them and allow for correlations?
It's like a multivariate regression for multiple outcomes. I think it may be from a statistical standpoint, that is something possible to do. We just haven't really looked into that in this project.
It'd be like a flock of penguins or something.
Our third speaker is Dr. Nicolau Martin Bassos from University of Bologna, who's also going to visit UW-Madison this summer. He's going to give us maybe a showcase of using PGS in social genomic studies, and the topic is the impact of mental health on labor market outcomes.
One second, let me see how this works. So, hello, my name is Nico. Seeing how it works. Thanks so much for inviting me to the conference. It's been amazing, seeing such interesting talks and just a bit of introduction about myself and about this paper. I'm an applied microeconomist. I work mostly on dementia-related topics during my thesis and age-related cognitive deterioration. Then at some stage, I come across with this kind of genetic data. I started getting interested into these topics, and I noticed that one of my supervisors, she had also this kind of interest, Sonia Deniu. She's also a co-author in this paper, and basically we took into this side quest to running this paper in terms in a parallel manner to the PhD. Then John Deniu, her husband, joined us for this project. And let's say we've been a bit slow because it was a side quest, and John and Sonia had a baby on the way, and then I moved to Zurich at some stage for a postdoc on a completely unrelated topic when Danusha joined. So long story short, we've been working for this in a while, but it's still very much a work in progress. So any comments are super welcomed, and so approach me or whatever afterwards. Super welcome to hear your thoughts. So the idea is actually pretty simple. I think it's nice for such a kind of short kind of session. It's basically in labor economics and health economics, we're often very much interested on how mental health affect labor market outcomes. And normally we kind of face these typical kind of reverse causality problems where it's unclear if poor mental health actually causes poor labor market outcomes or the other way around. No, basically, do I lose my job because I'm suffering depression and this makes me perform poorly, or is it the other way around? Do I lose my job and this triggers my depression? And the typical econ literature uses IVs, lots of these estimates are highly local, that is a problem or a limitation with when interpreting this. We noticed that this kind of approach of using kind of genetic markers for depression had not been used, and that's basically what we are doing. We are using polygenic indices to predict how individuals navigate throughout their labor lives when they face genetic predisposition towards depression, and we explore very much the gender dimension because we expect that there will be stark differences because there's important gender norms when it comes to mental health and labor market outcomes. And basically what we will find is precisely this, that females tend to work to hold much shorter tenures, while males kind of tend to shift towards less qualitatively dimensions of occupations, but they tend to work a similar number of years. So this is kind of consistent with this idea, that maybe there's a starker gender norm for males to be active at the labor force. So I am using the health and retirement study. I don't think I have to introduce it much because we've been sharing lots about this database in this conference. Basically, kind of two things I just want to mention is first, it's kind of old age household survey. So we are kind of talking about boomers, like an aging population. Second point, it's a US representative sample. We are interested in two pieces of information of the HRS. First, the genotype information. We will take the PGI for depressive symptoms. And then the HRS has actually very good information about labor market-related outcomes. We are particularly interested in the information related to labor life histories. So the HRS collects self-reported information about kind of labor life calendars. So individuals give information with lots of detail. It's more or less like 3-digit, I think, like census code levels or across 1,000 different occupations. We restrict our sample to European descendants and with non-missing information at the genetics and at the labor life histories. So what we do then, we merge the HRS with 3 different databases. Basically, we want to complement this kind of labor life histories with external information. First, the National Occupational Employment Wage Estimates. This is a database from the US Bureau of Labor Statistics, which provides you typical salaries in a large number of occupations, again at a 3-digit level code. So we basically match these labor life histories with typical annual salaries at different occupations. Second database we are kind of merging to the HRS is the occupational task data of David Dorn. Basically, at the 2013 AR paper, they released this database that provides descriptors of different tasks that are typically performed at a large number of occupations, and they focus basically at abstract occupations, which involve formal reasoning, managerial and interactive responsibilities, routine occupations, which are more related with cognitive tasks, and then manual occupations, which are basically level to eye to hand coordination. And finally, we merge this information of labor life histories with the OSTARnet. The OSTARnet is a database of the US Department of Labor. It's become very popular across labor economists in the last years, and basically it describes different dimensions of occupations, like values that are typical in different occupations, activities, and many other dimensions. We basically focus in work conditions, which is an aggregate measure including dimensions of activity, compensation, independence, and security. It gives us a sense of kind of better or worse work conditions in different occupations. And then we look at work characteristics. We have like 45 variables in the OSTARnet. We run a factor analysis. We end up with a group of variables that are a bit more easy to interpret, which we end up setting in, grouping in 3 different sets of work characteristics that are kind of descriptive of interpersonal relations, kind of this kind of more social communicative skills, then physical work conditions, which are related with kind of work ergonomics, environment, like job hazards, and finally, structural job characteristics, which are related with kind of the hierarchical structure of occupations, the level of the impact that individuals have on the final outcome, etc. So in terms of methodology, it's, as I was saying, it's a pretty simple paper at the moment. So we are running these linear probability models where we have these different outcomes like work duration, salaries and work conditions, work tasks and work characteristics. Then we have this interest variable, which is the PGI for depressive symptoms. Then we have like a vector of exogenous variables, basically year of birth, region of birth, gender, interaction with each others. And then we have also 10 ancestry-specific PCAs. And then to control for parental genetic effects, we also control this vector Z, which has like a parental socioeconomic status and parental education. Also childhood socioeconomic status and parents' education. So when looking at the results, basically I'm presenting you the results for the total sample and then a stratified sample by gender. So if you focus in column 1, like you can see, this is a more kind of saturated model. I'm not including still like this kind of childhood SES variables. You can see like I'm starting with work duration. I have several outcomes, but I start just with total years worked. If we look at column 1, this kind of saturated model, an additional standard deviation in the depression symptoms PGI can be or is associated with a decrease in more or less half a year of total years worked for the total sample. So interestingly, when we move at column 2, this, and we include this kind of SES controls, childhood SES controls, this estimate reduces by 20%, which indicates us that it's actually important to include those controls, and that's basically what we will do for the rest of the presentation. Now, the interesting part comes basically at these columns 4 and 6, where we see these kind of differences by gender, and here we do see like the coefficient for males is roughly like 1/3 the size of the one of females, and these stark differences are even stronger once we compare this to the mean. We see like the average years worked for males is more or less 12 years more than for females, so compared to the mean, this is a reduction in 0.5%, while for females it's 2%. So stark differences, and basically how we interpret it is we see these very strong associations between depression symptoms, PGI, and total years worked for females. We see 0 coefficients or small coefficients for males. You can see it's pretty noisy as well. So then we look like this at these other conditions, like as I was saying, salary and work activities. Basically, you can see like this, again, like massive coefficients for both males and females, and large differences. You can see like an additional standard deviation in this depression symptoms PGI is associated with a reduction of 1,800 annual US dollars for males. For females, it's roughly 1,000. Still, like, these differences have to be interpreted carefully because, like, compared to a mean, this is roughly a reduction of 3% compared to a mean for males and 2% for females. These differences start to become more apparent when we look at these. At the work conditions, where, for instance, this aggregate measure, that kind of captures better or worse work conditions, you can see we start seeing like there's much more action occurring at this kind of upper panel for males. We can see like an additional standard deviation in depression symptoms PGI is associated with a 3% of a standard deviation reduction in the work conditions for male. It's only 1% for females. And interestingly, when we move to the job task, you can see that for both males and females, there is this negative association, like kind of suggesting that both males and females, they move to occupations with fewer work tasks. But you can see that the coefficient is double the size for males compared to females. And interestingly, males additionally also seem to tend towards more manual and routine-based occupations, while for females we see this very clear zero coefficients. I'm not sure why this isn't working. Okay. Okay. Then, looking at these Osternet variables, I start looking at the interpersonal relations. So basically, we can see that for both males and females, like it looks like a higher predisposition towards depression is associated with less interpersonal relations. No, you can see like again like some coefficients, that can be interpreted as a 5% more or less of a standard deviation, and you can see like these individuals, they hold occupations with a lower load in communication skills, road relationships, conflictual contact, responsibility for others. But not much differences by gender. Same with the structural job characteristics. You can see like these individuals with high genetic predisposition towards depression, they tend to move towards occupation with more pace and scheduling. We don't see much for routine versus challenging work, and they tend to move towards occupation with less criticality of position, which is basically the impact of their job on the final kind of outcome of their activity. Finally, when we look at these physical job characteristics, here it becomes very clear. There's like these large differences across males and females. You can see these zero coefficients for females, if not negative, at some of these items. While for males, there is this very clear kind of positive trend, which suggests that males with higher genetic predisposition towards depression move towards occupations that are kind of worse in terms of physical job characteristics. The work ergonomics is worse, like the work setting, the body positioning, the environmental conditions are worse. There's more loud noises, they're exposed to dangerous substances, they're exposed to job hazards, they have to wear more work attire. So it looks like there's kind of stark gender differences when we look at this dimension. So in terms of robustness checks, we've done a couple of things. First of all, we controlled for educational attainment PGI, like EI2 and EI3. We also controlled for educational level, like as a variable, like the coefficients. In most specifications, they shrink in size, but these kind of trends we were observing, they remain constant. So to be honest, these are not our preferred specifications because we think it's a bit of an overkill, but we think it's interesting to see how kind of robust these results are. Then we also look at education as an outcome, looking in terms of kind of mechanisms. We add additionally PGIs to control for pleiotropic effects. We add PGIs for general cognition, anxiety, subjective well-being. Everything is pretty robust. We use the PGI for major depression. Everything is super robust in this case. Then we play around with the definitions of occupation. At the moment, we're basically looking at longest lifetime occupations. We look at the different definitions of this, and we also play around with our factor analysis, very robust results. And finally, we do this kind of thought experiment, like if we would consider these results to have a causal interpretation, let's see how much unobservables would drive those kind of effects down to zero. So yeah, we see this pretty robust results. So yeah, basically, as I was saying, we see that overall,
like a higher genetic predispositions towards depression leads towards worse labor market outcomes, but we see this kind of clear trends by gender. It looks like females, they tend to work fewer years, while males, that they hold work a similar amount of years, but they tend to shift towards dimensions that are qualitatively worse. This, as I was saying, is consistent with what we would think of the gender norm of that time, kind of pushing maybe males to be more active at the labor force, and maybe females maybe moving more towards unpaid occupations, like being housewives or this type of unpaid occupation. So yeah, maybe this is the last slide, and maybe that's the one that I'm more interested to present, because that's the next steps, and where I want to hear your feedback. So basically, at the moment, we are applying for social security records. We have like this kind of high-quality data of HRS wages. Basically, we would have like, for many individuals, their total labor life in terms of their wages, and we could explore this dimension of kind of chronological aging to see if these associations we've explored also occur constantly throughout time, or actually have this kind of differential kind of trends by aging. Second point we're exploring, I am personally very much interested in this literature that explores how technology shocks and how labor market shocks change the return to certain skills, to certain activities. And since we have the labor life histories of individuals, I would expect that individuals are moving throughout their lives toward work tasks, work activities that are becoming more or higher returns. And I would also expect that these individuals with higher genetic predisposition towards depression actually are less flexible, stay more in the same kind of work tasks that they've done throughout their whole life. So yeah, thank you very much.
Thank you, that was very interesting. So relating to the gender differences, did you show to us like whether the PGS was, whether its prediction would vary by gender?
Sorry. Like
did you show to us whether the prediction of the PGI, like its ability to predict depression, was different across men and women?
I have looked. I haven't showed it, but I have looked at it. Like it's the predictive power is stronger for females.
Okay, so that could partially explain why you're seeing this, right? So if you're doing an adjustment, that could explain why the reduced form effects in a way are larger for women. The other thing that may be really interesting to see if it's the depression PGI is related in any way to fertility. Is that the reason why women the effect may be on women, just because
yeah they move towards like
So I'm going to put back on the labor economics side. So given the time, so I was wondering, have you given any thought at all to what economists call labor supply on the extensive margin? So at the time, women, there's a lot of selection into who participates versus who doesn't. Is that controlled for at all?
The what, sorry? Like if they
choose, it's a choice-based sample for women, so are you accounting at all for that choice?
Of working, ever having work, like this kind of thing. Yeah. Also, we do have like a partner. Also, basically, if we are kind of conditioning on ever having work, is it kind of
or timing of work or something? So,
yeah.
So right now, implicitly, you're assuming
in the
it's
paper
the same
we have
process.
actually these two sections. Like that's a part of outcomes, like conditioning on whatever, no, like having worked or not, and the other like individuals who ever work.
And then two other quick suggestions. One is, so this is also for efficiency purposes, I'd pool the men and women together and put in an interaction term. I think it's a lot easier just to show the effect on the interaction term than forcing people to do ocular tests comparing things. This is my own. That's my own preference. And the other thing is, can you match this to like Medicaid data at all on prescription drugs? Because that way there you could actually see.
I guess we could I know with the social security records we're having some problems because we are already using this kind of restricted data, this labor life history, so it's a bit tricky to so I'm not sure if we will have similar problems, but that's something we could actually see like looking.
Let's thank our speakers again.
All right, well, welcome back everybody. Let's go ahead and get started. So our next session is our second session on epigenetics. If our panel could go ahead and come up.
And our first speaker is August Zuckerman from Drexel University, who's going to be presenting research on epigenetic aging moderates the association between lower socioeconomic status and faster declines in kidney function across a decade.
Good afternoon. I'm excited to share some of the work that I've been doing with MIDUS data, which we will learn soon. My name is Agus Rahman. I'm an assistant professor in the Department of Epidemiology and Biostatistics at the Dornsife School of Public Health, Drexel University. So I think I made this slide at midnight and I was tired. So it's not moderates, but mediates. So that's a huge mistake, but just, and I should change it, but well, I can tell you. To start, I would like to talk about chronic kidney disease and end-stage renal disease, which is a major public health problem right now in the United States, as around 15% of adults in the United States are expected to suffer from chronic kidney disease, which usually characterized by kidney damage and progressive decline in kidney function. And around 100,000 individuals are expected to suffer from end-stage renal disease, or the last stage of chronic kidney disease, which is characterized by kidney failure and the need for transplant and transfusion. So given how striking this problem is, one of the goals for the Healthy People 2030 is to reduce the burden of chronic kidney disease and end-stage renal disease. But one of the major challenges to achieve this goal is that there's evidence of socioeconomic and racial disparities in both chronic kidney disease and end-stage renal disease. Individuals from lower socioeconomic status and also those coming from racially marginalized background, they have higher burden of chronic kidney disease and end-stage renal disease. So I'm a developmental scientist by training, so and I like to think that looking at kidney disease from the perspective of developmental science, I think, give us a better understanding about the progression of this disease and how these disparities come about. So the blue line here represent kidney function of a healthy individual. As you can see, kidney functions start low when you're born, and then it reaches its peak during early adulthood, and it continuously declines as part of our aging process. But for most adults, they're not developing chronic kidney disease. The dashed line there is the cutoff for CKD based on our kidney function. Now contrast that to this orange line that represents someone who developed chronic kidney disease. So the progression of the disease itself is usually started with the stage of hyperfiltration to compensate the kidney damage, but usually followed by faster age-related declines in kidney function and end up in the disease stage or the stage of hypofiltration. So what I'm interested is to understand these differences in the progression of kidney function prior to the development of chronic kidney disease as a way to maybe find effort to intervene early so we can lower the burden of chronic kidney disease and eventually eradicate the disparities in chronic kidney disease. So as I mentioned earlier, lower socioeconomic status and minoritized racial background is associated with higher rates of chronic kidney disease and end-stage renal disease. But also those with lower socioeconomic status and coming from racially marginalized background, they have shown evidence of faster decline in kidney function, even among healthy individuals. And we know that faster decline prior to CKD and ESRD stage is a strong predictor for the development of chronic kidney disease. And it's usually evident among individuals from lower SES and racially minoritized background. So the main goal of this study was grounded by this idea of geroscience, just that there are hallmark of biological processes that underline the aging process. And epigenetic alteration is one of the hallmark of aging. As we get older, the epigenetic processes tend to be less stable, and that's why we've been seeing talk about epigenetic clocks, which was developed to predict biological age as relative to chronological age. And we've seen more studies coming out looking at the link between epigenetic aging, especially using the epigenetic clock measures with some of the age-related chronic disease that we usually see and pose as public health problems in the United States. So studies have shown that epigenetic aging is associated with higher probability of developing cognitive impairment and dementia, and also cardiovascular disease and mortality. But we haven't seen any studies try to look at the association between epigenetic aging and the development of aging renal function among adults. So that's exactly the goal of this study, to see whether epigenetic aging mediates, not moderates, the association between socioeconomic status. As we know, lower SES is associated with a faster decline in kidney function. And additional goal is to also explore whether this mediational relationship between epigenetic aging and SES and declines in kidney function was different across different racial groups among adults in the United States. So to test this hypothesis, we utilized data from the Midlife in the United States, which we will hear soon. We use data from wave 2 and wave 3 of MIDUS, which was 10 years apart. And we included participants who completed the biomarker portions of MIDUS study in MIDUS 2 and MIDUS 3, and also they have available serum creatinine information in both waves, and they also consented to the genetic analysis based on their biological sample. So another inclusion criteria was that they have to self-identify as non-Hispanic white and also non-Hispanic black. So in the end, we have analytic sample of 274 individuals between ages 37 to 83, and almost 1/3 of them self-identified as non-Hispanic black. So for socioeconomic status, we use composite measures of SES based on various indicators, including household income, education, insurance coverage, and also we included some measures of subjective perception of financial hardship. SES was measured in MIDUS 2, and then for the epigenetic aging measures, we calculated the age acceleration by regressing chronological age into the epigenetic age measures, especially for the first and second generation epigenetic clocks, including the Horvath, Hannum, PhenoAge, and GRIM-Age, both the version 1 and version 2. And also we have the third generation epigenetic aging measures, which is measuring the pace of aging based on the Dunedin pace measure. And all of these were measured during the MIDUS 2. And finally, for kidney function, we use the serum creatinine-based estimated glomerular filtration rate that calculated the proxy for kidney function, and we calculated those measures using the CKD EPI formula with no race adjustment in the formula. And we calculated absolute declines in kidney function between MIDUS 2 and MIDUS 3, which count for a 10 years period. On average, I think it's about 11 years between MIDUS 2 and MIDUS 3 follow-up. And we included covariates from MIDUS 2, including age at baseline, the years between the follow-up, and then also sex and some health-related measures that we know associated with probability of developing chronic kidney disease, such as smoking, obesity, elevated blood pressure, and also insulin resistance. So across a decade, kidney function on average declined on a rate of 10 milliliter per minute, which is consistent with the literature, so about 1 milliliter per minute. This is very normal for healthy individuals. But when we look at declines based on race, we see that non-Hispanic Black individuals in MIDUS, they have faster decline. And then we found that lower socioeconomic status was associated with faster declines in EGFR, as you can see on the graph. Those from lower level of socioeconomic status have the faster declines in EGFR, almost 15 milliliter per minute. And we found evidence of mediation by epigenetic aging, especially by the second and third generation of epigenetic clock, including the Grim age 2, and Danadin pace. This graph here represents the partial mediation by Grim age acceleration. So some of the association between lower SES and faster declines in kidney function was partially mediated by the second and third generation of epigenetic aging. When we look at the exploratory analysis on the differences based on race, we see that with the significance of index of moderated mediation by race for those 3 clocks that's significantly associated with declines in kidney function. And we see that the mediation by epigenetic clock was only significant among non-Hispanic white, but not among non-Hispanic Black across all those 3 clocks, as represented in this graph here. As you can see, among non-Hispanic white, the dashed line, higher epigenetic clock was associated with faster decline in kidney function, but not among non-Hispanic Black. So to summarize, we see that lower SES was associated with faster declines in kidney function across a decade, and we see evidence of epigenetic aging mediating this association to show evidence that this is one of the cellular biological pathways from lower SES to faster declines in kidney function. Regarding the non-significant association mediational pathway among non-Hispanic Black, we see that maybe this is due to the small sample size of in this analysis, but maybe this also would be, problem with the epigenetic measures that we have that may be not really sensitive to detect epigenetic aging among non-Hispanic Black, or maybe because epigenetic aging tend to be higher among these individuals across the board, across different levels of socioeconomic status, that we didn't see a lot of variations among this subsample. So the next stage is to, SES is one of the social hallmark of aging. There are other social hallmarks of aging, including stress, adversity. So the next stage is for us to look at how stress may be play roles in this relational association, mediational association between SES, epigenetic aging, and declines in kidney function as risk factors for chronic kidney disease and renal disease. With that, I would like to acknowledge the funding sources for MIDUS for myself, and also I would like to mention the co-authors for this paper. Yeah. Thank you.
We have time for one question, one quick question.
This probably reveals my ignorance about epigenetic clocks, but I'm just, if you could just help us think through a little bit. If you wanted to validate an epigenetic clock, would you validate it by looking at things like kidney function? This idea of that the aging is leading to the decline in kidney function, I'm finding it a little bit confusing because is the decline in kidney function essentially biological, the same thing as biological aging, or is there a difference there? if you could help educate us in that.
Right. I have a slides about explaining different generations of epigenetic clock, but I think the MIDUS workshop probably going to talk about that. But in general, as I mentioned earlier, there's this hallmark of aging that basically the biological underlying processes that associated with all the aging process that we see, and a lot of chronic disease that we're interested in our research, cardiovascular disease, chronic kidney disease, those are age-related chronic disease, right? Like there's a stronger correlation with age. And epigenetic aging, epigenetic clock was initially, the first generation, Horvath and Hannum, was initially developed to predict chronological age. How we can this measure how can they be closely correlated with chronological age. But the second and third generation of clock, I think they're developed this clock to really see whether they can also predict some of the aging phenotypes. Like the Grimm age, for example, is really designed to whether it can predict mortality. Right? So all the CPGs that they picked to calculate this algorithm to construct this clock was really developed to correlate with aging phenotypes, right? So Jonathan Pace, for example, looking at all these biological systems, and kidney function is one of them, one of the measures that they included in the construction of this clock. So I should probably explain that better. The reason why we see association with cognitive function, with cardiovascular disease, because those are the CPGs that I picked to calculate this clock were correlated with some of these aging phenotypes, including the disease that we care about in our research.
Thank you.
Thanks.
Next we have Lauren Rogers, who's going to present on epigenome-wide mediation analysis of the relationship between psychosocial stress and cardiometabolic risk factors in the health and retirement study. Welcome,
Lauren. Good afternoon, everyone. My name is Lauren Obsasnik Rogers. Just a quick name change. So, and I'm a PhD candidate at the University of Michigan. And today I'm going to be presenting some of my dissertation work entitled The Epigenome-Wide Mediation Analysis of the Relationship Between Psychosocial Stress and Cardiometabolic Risk Factors in the Health and Retirement Study. So psychosocial stress, which is defined as the consequence of an imbalance between adverse experiences that we meet in our everyday lives and our ability to cope with them, can be triggered by a multitude of stressors. So these include life events like job loss, discrimination, or financial uncertainty, as well as work-related or community stressors. So as many of you know, high levels of stress, particularly over long periods of time, are strongly associated with negative health outcomes, which include the development of chronic diseases like diabetes, hypertension, and heart disease. So in terms of the epigenetic impact of stress, prior research has found associations between stress and certain genes, and some of those genes have been found to play a role in the development of cardiometabolic disease. So we hypothesize that epigenetics may actually be the mediating link between stress and cardiometabolic disease, as well as its corresponding risk factors. So to answer this, or to explore this hypothesis, we came up with our main research question, which is, after adjusting for relevant confounders, does DNA methylation mediate that relationship between psychosocial stress and cardiometabolic risk factors? So to answer this question, we use data from the Health and Retirement Study. So in 2006, HRS actually added this psychosocial and lifestyle questionnaire, which captures information regarding participants' well-being, their lifestyle, and their social relationships. So our main exposure of interest, which is psychosocial stress, was measured either in 2010 or 2012, depending on which year the psychosocial questionnaire was actually administered to participants. And so here I have a timeline of all of the other covariates that we measured as well. So in 2016, that's when HRS invited participants to participate in an ancillary study, the venous blood study, or VBS, and that's where blood-based biomarkers were collected. And DNA methylation was actually collected in a subsample of those participants who completed the VBS study. And then our cardiometabolic risk factors were captured either during the VBS in 2016 or during the enhanced face-to-face interview in either 2016 or 2018. So after we excluded individuals who were missing data on any psychosocial stress variables, any DNA methylation or relevant covariates, we had about 2,700 participants who were included in our final analysis. So consistent with previous research that had studied psychosocial stress in both HRS and other cohorts, we examined 6 domains of stress across the life course, which you can see up here. So they included acute life events, financial stress, neighborhood stress, relationship stress, lifetime discrimination, and childhood adversity. And because we were interested in cumulative stress, again, across the life course, we created a summary stress score, again, based on previous literature, by transforming each of the 6 stress domains into z-scores, then summing the standardized scores from all of those domains and re-standardizing those sum stress scores. So this cumulative stress score actually is going to represent the total stress exposure across all domains. And then in terms of the cardiometabolic risk factors that we included as our outcome, we actually examined 10 cardiometabolic risk factors to assess that relationship between stress and cardiometabolic risk. So cholesterol, glucose, triglycerides, and CRP were collected in 2016 through the VBS, and then BMI, waist circumference, and blood pressure were collected during that enhanced face-to-face interview in 2016 or 2018. So here we have tables with some of our sample characteristics. The mean age of our sample was approximately 70 years of age, and we did have some variability in terms of race and education, with the majority of our sample having a high school degree as their highest level of educational attainment. Something else to note is that over half of this sample reported taking lipid-lowering and antihypertensive medication, while 20% reported taking diabetes medication. So this is a high proportion of the sample, and because of that, we did adjust certain risk factors like cholesterol, glucose, and blood pressure for medication use as we saw appropriate. So getting into some of our results, as a first step before we went into any type of mediation analysis, we wanted to assess if psychosocial stress was associated with each of those 10 cardiometabolic risk factors. So for our model 1, which is the left-hand side of the 2 results, we adjusted for age, sex, education, year of psychosocial interview, and top-gen genetic principal components. And we found, as you can see, associations between stress and BMI, waist circumference, diastolic blood pressure, HDL cholesterol, and CRP. And then additionally, as a sensitivity analysis, because smoking may actually be on that mediating pathway between stress and cardiometabolic risk factors, we further adjusted our model 1 for smoking to observe independent associations between stress and cardiometabolic risk, independent of smoking. And as you can see, we obtained substantively similar results from that. Okay, so then next for those 5 risk factors that we had found to have established relationships with stress, we then conducted an epigenome-wide mediation analysis to identify how much of those relationships were actually mediated by epigenetics, if at all. So to do this, we decomposed the total effects of the relationship between stress and the cardiometabolic risk factors into both their direct and their indirect effects. So the direct effect is going to be the effect of stress on each risk factor, absent of DNA methylation. So looking at this graph, the direct effect will be defined as that top arrow from stress to cardiometabolic risk, labeled beta XJ. And then the indirect effect is actually going to be the effect of stress on each risk factor that works through DNA methylation. So that'll be the middle path from stress to methylation to cardiometabolic risk. And what we're interested in is the mediation effect, which is going to be defined as the product of the exposure mediator, which is alpha J, that first stress 2 DNA methylation path, and then the mediator outcome effect, beta J, adjusting for the exposure. And you can see in the two equations on the top left-hand corner, those are the covariates highlighted in red of interest. And so we evaluated the epigenetic mediation effect by testing that null hypothesis alpha J times beta J equals 0 for J CpG mediators, which in our study ended up being about 790,000 after quality control. And we used the high dimensional mediation testing, or HDMT method. So here are the high dimensional mediation testing results for the association between stress and the cardiometabolic risk factors. So if you look at the second column, we found that the HDMT method identified 50, 46, 7, and 12 CpGs that mediated the relationship between BMI, waist circumference, HDL, and CRP respectively at an FDR corrected value of 0.05. And for each of those 4 risk factors, those individual CpGs mediated anywhere between 4 and 14% of that association between stress and their respective risk factors. And something to note, we did not find any CpGs that mediated the relationship between stress and diastolic blood pressure at an FDR Q of 0.05. We also were interested in determining that cumulative effect of the percent mediated by across all CpG mediators, but unfortunately, we do know that CpGs are not independent of one another, and the HDMT method could not account for the correlation between CpGs. So what we did is we used a principal components-based approach in order to approximate the cumulative effect of the CpG mediators. So in column 4, you can see that cumulatively, the methylation PCs that we calculated and then added together to get that cumulative effect mediated between anywhere between 31 and 46% of the stress risk factor associations. And then finally, for each set of the CpG mediators that we identified in the analysis, we used a de-biased lasso regression to identify CpGs that actually independently mediated that relationship between stress and risk factor after adjusting for all of the other CpG mediators. So columns 5 and 6 will detail those results. So you'll see the CpGs that were independent mediators, and they mediated anywhere between 3 and 13% of the associations. So many of the top CpGs from our HDMT method have been previously identified in other EWAS, which I have here in this table. So as you can see, there's similarities between CpG mediators across outcomes, where several of them are associated with chronic diseases like BMI, type 2 diabetes, heart disease, COPD, and inflammation as well. So additionally, to better understand the underlying pathways of the CpG mediators, we conducted gene ontology, or GO, in Kyoto Encyclopedia of Genes and Genomes, or KEG, pathway analysis to actually understand the biological implications of those CpG mediators. And we used 2 different methods for this. So first, we mapped each of the CpG sites using the annotation files to the nearest gene, and then we used that information for the gene set analysis. However, we do know from prior research that CpGs don't actually always act on their nearest gene, but may actually affect the expression of distal genes as well. So therefore, we used a separate database to map the CpGs to genes that we know that they affect the expression of, regardless of their location. And this allows us to discover potentially other pathways. So when we did map the CpGs to the nearest genes, we didn't observe enrichment of GO or KEG pathways for any of the cardiometabolic risk factors after FDR correction. However, when we did use the CpGs mediating the relationship between stress and each of the risk factors using the expression database, we found they were associated with expression of genes enriched for several GO terms. And I am showing these GO pathway results up here for BMI and CRP. So CpGs mediating stress and BMI were associated with the expression of genes enriched in pathways related to cell surface and cytokine binding and receptor activity. Multiple of those pathways kind of lead up to that general pathway. And then CpGs mediating the stress-CRP relationship were associated with genes enriched in those same pathways, as well as pathways related to neuron development, which I thought was particularly interesting given psychosocial stress is our main exposure. And then we did see waist circumference shared similar pathways to BMI, and HDL shared similar pathways to CRP. We had plots that looked very similar, so we are seeing consistency among outcomes as well, which makes sense because we did also find overlap with CPGs. So in terms of significance, this is a novel epigenome-wide mediation analysis of the association between stress and several risk factors in a multi-ancestry population of nearly 3,000 adults. We identified a subset of CPGs that mediated the total effect of stress on BMI, waist circumference, HDL, and CRP. And further, these CPGs were associated with the expression of genes enriched in pathways related to cytokine activity and neuron development. So in terms of next steps, I would first like to replicate these findings in an independent cohort of multi-ancestry older adults. And secondly, a lot of this research, because of the lack of data, epigenetic data in HRS longitudinally, these are more association studies. So I would like to measure both stress, methylation, and outcomes across multiple time points to actually be able to establish temporality within those relationships. So I would like to thank my advisor, Dr. Jennifer Smith, and my team from both University of Michigan and University of Wisconsin for all of their guidance on this project. Thank you very much.
I think so. We have time for one quick question.
Suppose your hypothesis is borne out, how might it change efforts to intervene in these diseases?
That's a great question. I mean, I think that we already kind of assumed that the pathways between stress, like we knew the established relationships between stress and all of these cardiovascular and cardiometabolic diseases. I think this research now is just trying to understand those pathways. A lot of times when people talk about stress and cardiometabolic disease, they think of that pathway through inflammation. That's like the number one thing. But I think that now seeing potential other pathways, getting down to the genes, and then also potentially neuron pathways, it's just more understanding the biological relationship between the two. I don't know if it necessarily would change anything in terms of how you care for the disease. I think if anything, it impacts how you treat the exposure, so how we actually treat stress and at what point we intervene on stress, whether that be earlier in life or before this accumulation of stress affects those outcomes. So I don't think it necessarily will change the outcome, but potentially more emphasis on the mental health and the exposure.
Thank you. Thank you very much.
Finally, our last talk of the day in the conference, Arthi Bhatt from the Pennsylvania State University, who's going to be presenting The Great Recession and Epigenetic Aging Among Midlife and Older Adults.
Hi everyone, I'm really excited to be here today to present some of my work on as Lauren said, the Great Recession and Epigenetic Aging Among Midlife and Older Adults in the US. And I'd like to thank my co-authors, Abner Apsley, Dave Almeida, Steve Cole, and Idan Shalev for their help with this project. I'd also like to start off by acknowledging the T32 that I'm on through the Center for Healthy Aging at Penn State, as well as all the funding sources that make it possible for us to have the MIDUS data collection and support through the Penn State Population Research Institute. So to set the stage for the 2007 to 2009 Great Recession, the recession saw significant impacts across a variety of different domains, including financial market impacts that saw GDP declines of 4.3%, stock market prices fell to less than half of their previous peak, and the median net worth of families dropped by about 40% during this time period. Additionally, there were substantial labor market impacts due to the recession, in which the unemployment rate increased from about 4% to 10% during this time period, and the poverty rate spiked to 15% in the United States. And finally, the housing market impacts of the recession included declines in home prices by 33%, over 15% of mortgages being delinquent or in foreclosure during this time, and homeless shelters in certain cities of the US seeing an increased intake of up to 40%. So there's been some work previously on the impacts of the recession on health outcomes, specifically physical and mental health, such that exposure to recession hardships has been shown to be associated with increases in depression, anxiety, poorer rated mental health, suicide, and substance use, as well as physical health issues, including increases in poor self-rated physical health, higher levels of chronic conditions, and allostatic load. So as we've been talking a little bit about the past couple of days, life's experiences and stressors can contribute to DNA modification, such as DNA methylation, which is when a methyl group is added to most typically a cytosine, followed by a guanine base in DNA. And these DNA methylation patterns can affect gene expression by potentially dampening or amplifying the expression of genes. So in terms of epigenetic aging, these DNA methylation patterns at the cytosine-guanine base pairs, which we'll refer to as CPG sites, have been shown are associated with certain markers of aging. And these have been used to develop epigenetic clock measures, which estimate somebody's biological age. So as Agus had mentioned, and other talks as well, there are multiple iterations and generations of these clocks thus far. So the first generation clocks were trained to predict chronological age, including the Horvath and Hannum clocks. However, the more recent second and third generation clocks were trained to estimate biological age in order to better predict health span and mortality, including the pheno age and grim age clocks, as well as the Dunedin pace of aging. So in terms of calculating epigenetic age acceleration, for all of the clocks, with the exception of the Dunedin pace, the epigenetic age acceleration is calculated as the residual of somebody's biological age, which would be estimated by the epigenetic clock on their chronological age. And epigenetic age acceleration would be when there's an older biological age relative to somebody's chronological age, which is generally denoted by positive values, whereas epigenetic age deceleration, or when there is a lower biological age as compared to a person's chronological age, is denoted by negative values. However, for the Dunedin pace of aging clock, a score of greater than 1 indicates a faster pace of aging, and a score of less than 1 would indicate a slower rate of aging. And so, as we've talked about during the conference, epigenetic age acceleration is associated with a variety of different health markers, including chronic conditions and mortality. So there are also some significant race, ethnicity, and sex differences in epigenetic aging. Regarding race and ethnic differences in epigenetic aging patterns, these are more variable across different clocks in that so far we haven't really seen consistent patterns across different racial groups in terms of faster rates versus slower rates of epigenetic aging on these clocks. However, generally, females tend to have slower rates of epigenetic aging compared to males, which has been consistent with the trends that we're aware of in aging, that women tend to have longer lifespans compared to men. So regarding the recession in epigenetic aging, while there has been some prior studies that have illustrated a connection between economic hardships and accelerated epigenetic aging, there have been very few studies that have examined how these economic hardships that are experienced either in the context of recessionary events, such as the Depression or the Great Recession, and Lauren Schmitz has some great work on the Great Depression and epigenetic aging, how these hardships that might be experienced later on in the life course rather than earlier, because there's been a significant amount of work looking at in utero, earlier in childhood, or even in adolescence, but not as a growing body at this point that's starting to look at how hardships experienced in midlife might affect people's epigenetic patterns later in the life course. How these might look across different types of economic hardship domains and across multiple generations of epigenetic clocks in being associated with accelerated epigenetic aging. So the data used for this study was the Midlife in the United States study that Agus had mentioned and some folks at Minnesota are going to be talking about later today. But essentially this study started data collection in 1995, and it has 3 longitudinal waves at this point. However, I used the cross-sectional refresher wave, which started the data collection for the survey sample in 2011. And this is because participants provided information on a list of recession hardships in both the MIDUS refresher and MIDUS 3, but we only have the DNA methylation data at MIDUS 2 and the MIDUS refresher. So because of that, we decided to use the refresher cross-sectional sample for this study. And participants who provided information for the survey were also invited to provide biomarker data and provided whole blood samples that were then assessed for DNA methylation patterns to create the epigenetic clocks of interest. So overall, our sample size was about 750 adults with a mean age of 50, and about 18% of the sample were Black, 10% was other non-white race, and 52% of the sample was female. So for our measures, our independent variables of interest were recession hardships, and one of the main variables was looking at overall recession hardships across a list of 18 different negative recession events that participants reported either experiencing or not experiencing during and in the aftermath of the recession. So about 83% of participants reported at least one recession hardship, with a mean of about 3 recession hardships across the sample. And then in the individual domains of recession hardships, 78% had one or more financial related hardships, which could include responding affirmatively to an item such as increasing debt during and in the aftermath of the recession. About 37% had at least one job-related hardship, such as losing their job during or in the aftermath of the recession, or having to take on another job to make ends meet. And 35% reported one or more housing hardships, which could range from factors such as missing a rent or a mortgage payment, all the way to experiencing eviction. And our dependent variables of interest were the epigenetic age acceleration scores that we calculated, as I mentioned previously, from the 5 clocks of Horvath, Hannum, pheno-age, Grimm-age, and Dunedin pace of aging. And finally, the covariates that we used in the study included sociodemographic covariates of race, sex, marital status, education, household income, and BMI. So for the analysis, utilizing the linear regression, we tested associations between the overall and individual domains of recession hardships with our accelerated epigenetic aging scores, controlling for the previously mentioned covariates. We also created interaction terms between the overall and individual domains of recession hardships with both race and sex, and then repeated linear regressions to assess potential moderation effects on epigenetic age acceleration scores, controlling for the same list of covariates. And our results were reported as unstandardized regression coefficients, and we utilized multiple R squared to indicate effect sizes, and the alpha that we utilized was at a 0.05 level. So these were our results for the overall number of recession hardships. So as we can see, experiencing more recession hardships was associated with accelerated epigenetic aging across all of the clocks, with the exception of the Hannum. And we also saw interesting sex-based patterns, such that females had lower rates of epigenetic aging acceleration compared to males, which again is in line with previous work that has indicated that females tend to have slower declines in some of these areas compared to men. And we then separated out the analysis looking at each of the individual domains of these recession events. So we see here that the associations between the recession hardships and these epigenetic age acceleration scores seems to be primarily driven by financial hardships, and that financial hardships is associated with accelerated aging for all of the clocks, with the exception of the Hannum and Dunedin pace of aging. For job-related hardships, there were no significant associations between these hardships and our epigenetic age acceleration scores. And for housing hardships, there was only a significant association with Dunedin pace of aging compared to any of the other clocks. We also, as I mentioned, did some moderation analyses, but we didn't find any significant moderations by race. We only found moderations by gender at a p less than 0.1 level, such that for the Dunedin pace of aging, as participants in the sample experienced more job-related hardships, females tended to have a higher pace of aging compared to males. And for the Hannum acceleration scores, as participants experienced more housing-related hardships, females actually had lower rates of epigenetic aging compared to males. But again, given that this was at a 0.1 level, we're not thinking that these effects would be too significant in the results. So overall, recession hardships were found to be significantly associated with our epigenetic aging acceleration scores. Among individual recession hardship domains, financial hardships seem to be the ones that mainly drove this association, followed by housing hardships, whereas job-related hardships were not significantly associated with epigenetic age acceleration patterns. There was no significant moderation by race across overall and individual recession hardships, but there was some evidence of moderation by gender, again, at that p.1 level. So limitations of the study include that MIDUS doesn't provide information on how many times or temporally when these recession hardships were experienced. Participants simply report in a binary whether they did or did not experience each of the events during or in the aftermath of the recession. Also, due to the cross-sectional nature of the DNA methylation data in MIDUS, we were unable to control for any DNA methylation indicators at a previous wave to address endogeneity issues. And finally, this analysis is among the first that examines how recession hardships experienced in middle adulthood across multiple hardship domains as well might be associated with accelerated epigenetic age patterns across multiple generations of epigenetic clocks. And it also indicates a need potentially for geroprotective interventions for economically vulnerable aging adults in order to improve their healthy aging. And finally, future research should examine how the COVID-19 related economic hardships, which we'll be hopefully getting that data pretty soon, experienced across different points in time of the life course might be associated with epigenetic age patterns. And yeah, that's what I have, so thank you all so much.
We have time for a couple questions.
I was just curious, as you were presenting, do you have data on, I guess we'll find out after 2:30, about sleep patterns? Because I can imagine different hardships would affect sleep differently, and that could be
That paper is actually under review right now that I'm
not the
working
referee
on.
then,
Yeah.
okay.
So, but yes, in short, both across subjective aging, subjective sleep recollections, as well as using actigraphy measured objective sleep markers, these recession hardships are significantly associated with sleep.
So I think, so this is a genetic conference, so using the epigenetic marker is, you know, I can understand why you're using epigenetic aging, but epigenetic aging measure is in the discussion of the biological aging, and there are a lot of other markers of the biological aging, and why did not use other biological markers, but why your focus is on the epigenetic aging?
Yeah, we've been doing some work, looking at economic strain as well with the conserved transcriptional response to adversity, gene expression scores that are available through the RNA data in MIDUS, and also doing some work looking at hardships, specifically housing-related hardships in the aftermath of the recession and allostatic load patterns across multiple different physiological systems that are in that overall allostatic load score. So in short, yeah, we are looking at those as well, but for the sake of kind of this presentation, just narrowed it down to these epigenetic age acceleration markers. So thank you.
Thank you so much, Arti.
So let's get back situated and settled back in for a little overview of all the opportunities and ongoing work. Introduction to MIDUS. I'll let Bob introduce his Minnesota-based team and give us a little bit of a roadmap of what we're going to be doing.
Thanks.
Welcome.
Yeah. Thanks. So thanks, Jason, for organizing this meeting, which is great. Right? It occupies this important intellectual space I don't think any other meeting is quite sitting at, so that's excellent. And for the opportunity to present on MIDUS. So when this request was made of me, I thought, well, I'll just bring everybody along, because why not, right? So that's
Christian Marcon over there and Colin Freilich, and together we all work on MIDUS stuff at Minnesota, which is where I'm appointed. You may know that MIDUS actually runs through here, right? It runs through the University of Wisconsin, but we work at Minnesota. So without further ado, you've seen this before, right? So in the previous presentations, you've seen kind of how MIDUS is organized, but it's really been kind of a remarkable thing to have the privilege to be involved with over the years, right? So I got involved in MIDUS in the late 90s when I was an assistant professor, and it was because they were looking for somebody who was interested in twins. And I've been interested in twins throughout my entire career, right? And so I was fortunate to know Carol Riff. I got my PhD here, actually. And she was looking for someone to sort of deal with the care and feeding of the twins in MIDUS, and that's how I got involved. And then, you know, subsequently, it's become more molecular in its focus. So I helped run the genomics project within MIDUS along with Steve Cole, whose name you've seen before. Steve handles more like the wet lab stuff, and we handle more of the gen epi kind of stuff at Wisconsin. At Minnesota, which is where I actually work now. So overall, how is MIDUS structured, right? So there was a longitudinal part of MIDUS that you can see in this slide, right? MIDUS 1, funded by the MacArthur Network, right? Which then became MIDUS 2, which was the first NIA-funded MIDUS endeavor and adds, you know, other elements to the overall picture, right? Including subsamples. So as many of you may already realize, based on what you've seen already, the way that MIDUS works, right, is there's a large national sample, and then people are kind of siphoned off into more specific sub-projects. So for example, for the genomic data we'll be talking about today, those are people who participated in the biomarker subsample. So that can be confusing to people who are new to MIDUS, right? But you can imagine it from a budgetary perspective. You can't take a, you know, multi-thousands of person national sample and, you know, image them all, right? So you have to kind of like make do with pulling people into some of these subsamples. But it's a remarkable longitudinal study, as you can see at the top of this slide, right? So you see MIDUS 1, 2, and 3, and now we are fortunate to be funded for MIDUS 4, which is ongoing currently, right? So that's also portrayed on the slide. Between MIDUS 2 and MIDUS 3, there was an effort to refresh the sample, right? So to add new participants, so that's the MIDUS Refresher that's down there, and MIDUS Refresher 2 is even a continuation of that. But wait, there's more. In addition to what's on this slide, right, which isn't even featured on this slide, is MIDUS in Japan. So there's a parallel study, very similar measures to MIDUS that was conducted in Japan, that allows for some cross-cultural comparisons as well. So that's the basic overview of MIDUS. This slide, you know, is readily available on the MIDUS webpage. So let's get into what genomic data are currently available. I think throughout the 3 talks, we'll probably be talking about things that are related to each of these sort of omics, if you will. So the way to think about MIDUS's genomic data, or at least one way to approach it, right, is to say, well, which kinds of, you know, commercialized omics, you know, were applied, you know, in the people who were participating in the genomic subsample, which is basically a subsample of the biomarker sample. So for DNA, right, so, you know, genome-wide SNP arrays that have been imputed to the genome, right, so you can do things like polygenic scoring. If you don't include the Japanese sample, you have about 1,200, 1,300 persons, right, they're midlife, consistent with the title of the study. And there are actually a handful of people from MIDJA, the MIDUS in Japan study, who have been genotyped. That's portrayed in that first part of the slide there. DNA methylation, right, which you heard a little bit about in the previous, you know, set of talks. That's available on about 1,300 people as well. Are they the same exact people? Maybe not, right? So that gets confusing a little bit with MIDUS 2, right? In addition to the complexity of all the different subsamples, not everybody's participating in everything at the same time for the same reasons, right? This can be discerned to a certain extent by just running crosstabs on everything that you've, you know, downloaded, which we'll talk about in a minute. But there can be complexities to this because it's also continually evolving, right? So, you know, every once in a while you'll find that we've added something to the data set, and this is ongoing, as I showed in the previous slide, right? So as we're sitting here talking, MIDUS is collecting data, right, which eventually filters its way into the public release data sets. So yes, 3 omics, right, the DNA that I just mentioned, the methylation arrays, right, so the EPIC array that everybody's familiar with, And then, somewhat unique for sort of a large population study are expression arrays that were run. So, right, so you have genome-wide expression transcripts available from peripheral blood mononuclear cells that were collected, excuse me a moment, they were collected as part of the biomarker study. So those data are available as well. So yes, although this is sort of daunting from an analytic perspective, there are certainly cross-omics possibilities that exist here, right? So imagine something like, oh, I don't know, trying to figure out segments of DNA that might affect the expression of a particular transcript or something like this, right? So such things are in theory possible, right? The limitation, of course, for such discovery efforts, you know, net of false discovery rate and other kinds of considerations, is the sample size, right? So this isn't set up to be, you know, a de novo discovery kind of sample, in my view, given the size, but it's well suited to working with things that already have an established effect size. So, you know, I don't think I brought this slide, but I enjoyed making it, right? I did a slide which was just, you know, like, what's the predictive validity of all the polygenic risk scores that we computed in MIDUS? EA3 is the most predictive score. It's better than anything else, right? It's better than, like, lipid proteins and things of this sort. It's really remarkable. I think the Pearson correlation must be about 0.35, just the raw correlation between, you know, EA3 polygenic index and, you know, years of education in MIDUS. So, right, if that's really the close to the population effect size, perhaps debatable as to what the population effect size really is, but, right, like if we say that that's not an unreasonable thing to think, well, you know, you can detect it in a sample of this size. And I'll get into how, you know, you can, one could also use MIDUS as part of larger discovery endeavors. Let's say you wanted to use it, as people have, right, in the previous talks. Pretty straightforward. Just go to the website, midus.wisc.edu. The rest of that URL just, you know, is from a drop-down in the main URL. But if you just go, well, you could do it right now if you have an internet-connected device, to midus.wisc.edu, you can kind of see how the website is organized. But part of how MIDUS works, and this has been true for, you know, as long as I've been involved in the study, is that it's very important to treat it as a kind of a public resource, right? So it's not intended to be just for, you know, people like myself who are fortunate enough to be involved in the design of the study and that kind of thing, right? It's meant to be a widely available, widely accessible data set. And it is, it has been for some time, I believe, the most downloaded data set at the ICPSR, right? So a lot of people use the data, and that's the intent, right? To create kind of a resource for the field. So hopefully it can serve as a resource for, you know, work in social genomics, as we've been talking about at this meeting. So what's publicly available, like you could go get it right now, right, are summary scores, maybe not surprisingly, right? So there are a bunch of polygenic risk scores, right, that are available, you know, with documentation about how they were computed. What did I do, right? I managed to figure out how to send the data to NBER, right, so that ISU, who's a person I know, could compute the SSGAC PGIs. That's a lot of letters. But anyway, right, you guys know what I'm talking about here, right? There's an effort in SSGAC to kind of harmonize polygenic indices, and MIDUS is part of that effort. And I should probably email her to check in on that, right? But anyway, like she's got that stuff, and so supposedly at some point in the near future, those polygenic indices that are, you know, commensurate with what, you know, SSGAC is doing should be available in MIDUS. So that's that part of the slide. As you heard about, right, there are epigenetic age acceleration variables, so those are computed variables based on DNA methylation, these clocks that people are talking about. There's also a computed variable based on expression called the conserved transcriptional response to adversity, right? This is a concept that Steve Cole, my collaborator on this, has written a lot about. And so that's available, you know, readily right off of the publicly available data. Now, for this kind of sophisticated audience, right, you guys probably don't just want to use off-the-shelf scores, right? You want to do your own thing. And that does happen, right? This has happened to me more than once, right? I try to publish something with polygenic risk scores, and someone's like, you know, last month somebody invented the 9th way to do the polygenic risk scores. You better do it or we're not going to publish the paper, right? So when that happens, like, you need the raw data. And we do make the raw data available. So there's a special process, but it's not meant to be onerous or unpleasant, right? So there's a form to fill out, and it's mostly about just making sure that the data are secure at your, you know, facility and having kind of a data transfer agreement in place, and MIDUS tracking, you know, where the data are, you know, housed around the world. So yeah, the raw DNA, the raw methylation, the raw expression transcript data are also available. There's a request form that's sitting on the web page. It's not impossible to get them. We don't want it to be difficult to get them. I sign off on these things, right? And I'm very liberal with trying to give away the data, with good reason, right? Because the idea, as I was saying about MIDUS, right, is that hopefully people will use the data set, and it continues to be, you know, a valuable resource for the whole community. Oh, look, it's a book that I was a, you know, co-editor on. If you're interested in MIDUS, you might be interested in the book that Carol and I published. So Carol Riff is the PI of MIDUS, and she and I thought to put together kind of a compendium of, you know, different things that people had done with MIDUS over the years. So this was in the last couple of years. This was published, last 5 years or something like this, right? The Oxford Handbook of Integrative Health Science. So if you find MIDUS intriguing and your bookshelf is lacking this particular tome, right, you can always add it. But in all seriousness, right, that contains a lot of descriptions of, like, what is MIDUS, how are the data collected, what kinds of projects are people doing with it. So if you want a bigger feel for all the things that happened in MIDUS over the years, this might be a resource for that purpose. But what about twins, right? I mentioned twins originally. So yeah, this conference has been very molecular in its focus, but I did want to say some things about twins, because in addition to everything I just described, there's twins in MIDUS. So why do people still care about twins? Well, I mean, I do, right? Like, I still think that the twin study approach is just extremely valuable, so I just wanted to share some of my ideas about this by using a particular MIDUS example, right? So I'm a psychologist, right? I was trained, in fact, as a clinical psychologist, and I work in a psychology department, and when you sort of learn this stuff, you know, as an undergraduate in the textbooks, you find out, oh, like, twins are, you know, similar, and so that's some, you know, prima facie evidence for the heritability of human phenotypes, right? But less well publicized is the fact that you can understand, I think, causality better by working with twin data, right? If you can't manipulate things, you're kind of left with, you know, terrible things like instrumental variables or whatever, but you could really try, you know, to use things like twins, right, to also kind of leverage a kind of counterfactual account of causality, right? So you can think about twin studies as co-twin control studies, and that can hopefully provide some interpretive leverage in things like behavioral epidemiology. In particular, like, one thing that I think is important about twin studies has to do with biological correlates. So this happens all the time, right, in the literature that we're talking about in a meeting like this. What's the meaning of an observed correlation between a putatively biological variable and a putatively social variable? Well, that's ambiguous, right? If you just have cross-sectional data and you just computed a correlation, it's hard to interpret the meaning of that association. That could reflect shared antecedent causes, as everybody in this audience knows, right? Like, you could have pleiotropy, where genetic effects lead to both A and B. But it could also reflect something more like direct environmentally mediated causality, where A is causing B, right? If you're just observing a correlation in cross-sectional data among unrelated persons, it's hard to discern the difference between those two, you know, population models. So again, my, you know, sort of, I guess, assertion here is twin studies framed this way help with interpreting observed correlations, because they're going to better approximate a counterfactual account of causality when there are things, oh, like SES, a major topic at this meeting, right, that aren't amenable to experimental manipulation. And that's because twins are matched on at least their DNA nucleotide sequence. Things like their objective rearing environment, their sort of obligatorily shared rearing environment, if you will, their age, their gender for same-sex pairs, like that kind of thing, right, is matched. So there's a bunch of unmeasured, or not necessarily needing to be measured, let's call it, like, confounders, right, that are matched because of the way in which MZ twinning works, and that might provide, you know, some leverage on, you know, kind of a more interpretable causal-like effect, right, than just unrelated persons. oh, imagine this, right? Like, see, even if they're matched on the DNA, they could be different on their epigenetic markers, couldn't they? See, right? So there's stuff like that, right, that's kind of lurking in the data set that's, I think, interesting to think about. So I'll just give you guys an example. This is a paper it's been around for a while now. That's funny. Time flies. I feel like I just worked on this. This is a sample of MIDUS biomarker twins, and you can see the N is pretty modest for this kind of thing. So for those familiar with, like ACE modeling, right, the kind of classical biometrical approach to twin decomposition, you would have low power in a sample this size to distinguish additive genetic effects from shared environmental effects. But there's reasonable power to distinguish sort of between pair effects from within pair effects. There's power analyses in this paper that sort of point that out. But so even in a modestly sized twin sample, like there's some ability to distinguish between from within effects. And that's kind of what we had in mind in taking this approach here, right? So here is education predicting allostatic load, right, which is kind of an index of overall health or lack of health. And MIDUS has all this richness of biological measurements. So there's actually sympathetic, parasympathetic, and so on and so forth, different indices of overall physical health that are summed to create an allostatic load score here. So again, the basic idea is like we know that lower education is associated with worse health. Like that's one of those like hard to destroy observations in epidemiology that's hard to make go away even if you throw a bunch of covariates at it. But what was a little bit unique here, right, was the measurement of health through this more sort of intensive allostatic load characterization of, the health phenotype, if you will. A little bit of math never hurts on a Friday afternoon, right? This is just, the regression equation. But when I teach this stuff, right, I think this is a good way to point out kind of like where the interpretive leverage is sitting, right? So if you look at the first equation, well, that's just your standard like OLS style regression equation where you have some dependent variable is some intercept, right, plus some slope times, a predictor score X plus some kind of, residual or, disturbance term, right? So that's just your standard OLS regression. That's the reason I presented it this way, right, is because that's what you're limited to if you have people who are not related, right? If you just have a bunch of unrelated people and you have cross-sectional data, that's what you can do with two variables. Now, okay, you could throw in some covariates and things like this to try to get more at something that has greater interpretive heft, but if you have twins, I think you can do considerably more. I think that's a fair assertion to make. So if you look at the second equation, right, you just set it up as a mixed kind of model, right? So what you have now, right, is the outcome that you're studying, right, is some intercept plus the within pair effect, right, which is the person's score minus the within pair mean, right, that's that second term in the equation, plus the between pair effect plus the disturbance or residual term. So the point is, like, that stuff that's in the middle of the first equation can be elaborated, right, if you think about it this way, right? You can't unpack the stuff at the top if you don't have people who are twins, right, who have a certain degree of relationship with one another. But if you do have people who are twins who have a relationship with one another, the model at the bottom is estimable, right? And so that's, I think, the key thing to maybe appreciate here is that you can't estimate the second equation if you just have people who are not twins. And twins are not the same within pairs, right? So the leverage here, right, this is an important point, right? Like if the MZ twins are too similar, then this approach won't work, right? If something is too heritable, like height is not a good thing to try to study in terms of within pair effects because it's too heritable. But for things like educational attainment, at least in this sample, there were, a number of pairs who had different levels of educational attainment, suggesting that there's some leverage there, there's some variance, right, from the within pair mean. So that's plotted as the within pair educational deviations from the within pair mean. Just to make the point that the twins are not necessarily the same within pairs, suggesting, sort of prima facie evidence for some environmental effects, net of the, unmeasured confounders, right, that the twins have in common. So what did we find here? There are about, maybe like half the pairs were discordant on education. And interestingly, that that P is 99, right? Like this is the smallest effect maybe I've ever published. That could be true, right? But the within pair effect, there's no evidence for that being in any way appreciably different from 0, whereas the between pair effect was nominally significant here, right? So the entire phenotypic correlation, at least in this sample, between allostatic load and education was mediated through the between pair effect, right, which suggests that the phenotypic correlation between education and allostatic load is attributable to antecedent causes that are in common, which could be something like pleiotropic endowments, rearing environments, and there's no evidence here for direct environmental causation. So again, I think that kind of approach is important because if you look at the literature on education and health, it's routinely interpreted as if we improved people's education, we would improve their health. Right? This suggests that that's not necessarily how it works, right? Because you wouldn't know, right, how these things are connected if you didn't have some additional leverage to interpret it through doing something like this kind of twin approach. So there are twins in MIDUS. This study was done with the MIDUS twins. And so that's also an aspect of the data set that's available. Here's an example of working with the DNA. Again, like at this part in the talk, I'm just going to give you some examples from things we've done recently, maybe to like, you know, whet your appetite or give you ideas of things you might want to do with the data set. So here's something we did where we were looking at discrimination and anxiety. So this is the MIDUS biomarker sample participants looking at the European ancestry people for purposes of ensuring that the polygenic index had some predictive utility. And what we were interested in here, right, is the connection between discrimination experiences and experiences of anxiety. So there are these nice, David Williams, is part of the study, right? David Williams developed these nice measures of discrimination experiences, daily discrimination, lifetime discrimination, and job discrimination are all used in this particular study. And the idea, again, right, is, okay, well, can we predict anxiety from discrimination? And as you can imagine, that's another one of these like interpretively ambiguous things in the literature, right? Is it the case that people who are more generally sensitive, anxious, stress reactive, and so on perceive greater discrimination in their ambient environment? Or rather, is it the case that people who in a bona fide way experience these things end up with higher levels of anxiety and greater sort of mental health consequences? So, we did something like this, right? So you can see the covariates set at the top, right? So there's the usual kinds of demographic covariates on the left. There's the principal components, right, which people in this audience would be familiar with, right, from the eigen decomposition of the SNP data, and then polygenic risk scores for anxiety that were available in the literature at the time. This is not fully satisfying, and I completely appreciate that, right, because the polygenic risk scores are not particularly potent, right, as controls, but the logic, I think, is sound. Right, similar to the MZ co-twin control logic, right? What you're trying to do here is control for, genetic variance in trying to see if you can eliminate the, association between discrimination on the left side and anxiety on the right side, right? So that's the, path in the middle of this, structural equation model, with discrimination and anxiety having multiple indicators, right, as it's shown on here, right? So the idea is if you took into account all those confounders, can you, reduce the direct path to non-significance? And we could not here. Again, like the thing that's, unsatisfying, right, is that those PRSs are not particularly predictive of anxiety. But at least based on the modest predictability that exists at that time, that didn't eliminate the effect, that we observed, the association, if you will, between discrimination and anxiety outcomes. We heard a little bit about the Great Recession. Hopefully that never happens again. What is the dismal science doing about, bigger issues in our economy? Well, maybe that's a question we can talk about later. But in any case, right, there's this natural experiment, right, in MIDUS, right, of studying people who crossed that, particular episode, right, that secular change in our society. So another thing we did was just look at the impact of the Great Recession on mental health, financial, job-related, and housing kinds of recession impacts were measured explicitly in MIDUS. So not just that they were in that time period in terms of how they were being studied, you also know how it affected them directly because there were questionnaires specifically about those kinds of experiences. And I've always been interested in kind of mental health in secular context, right? Again, I mentioned I'm a clinical psychologist by training, so mental health is a big interest of mine. assessments here, past year mental health outcomes, and interview for major forms of what you might call internalizing disorders. So major depression, generalized anxiety, panic disorder, those are highly correlated with each other, which gives rise to the idea that they're part of a systematic spectrum. And what you see in MIDUS and many other data sets is most of the effect, in this case, it would be on recession impacts on internalizing disorders, is mediated through the variants that those things have in common. So that's been one of these things that I've been interested in throughout my career, right? That there's a lot of comorbidity among mental disorders, and what that means to my way of thinking, right, is that they belong together in these kind of coherent spectrums of variation. And in fact, in this particular paper, most of the effect of the recession impacts on internalizing were mediated through the internalizing variable, although we also looked at alcohol and substances. This is just a descriptive graph or figure from the paper, just showing that the number of recession impacts was related in a roughly monotonic fashion with the kind of mental health outcomes. So yes, I mean, again, ambiguous in terms of causality, but nevertheless notable, right? It's not really good for people, for the economy to be chaotic or problematic, right? So it would be good to get that under control. And mental health takes place in this kind of context. So see, this is a thing that's important to me for various reasons, because the world of mental health research, at least in the United States, is dominated by NIMH funding, and there's a strong emphasis on biological psychiatry, right? And sort of social psychiatry, if you will, which isn't even really a term in the US, that's like a European term, has received short shrift. But like people exist in a social environment, and I think psychiatric disorders occur in a social environment. And so this is, again, more evidence of like the broader social environment in which people exist is clearly relevant to their mental health and functioning. Okay. So here's another MIDUS example. Again, these are just recent papers that I've been involved in to kind of whet your appetite. So this is one that we did on the longitudinal prediction of mortality risk. This is just looking at the whole, larger sample. Originally studied in the mid-90s and followed through 2015 for all-cause mortality. And the question here was, what's predicting mortality? Is it a specific form of internalizing psychopathology, or is it kind of the common spectrum? So we fit a hazard kind of model here to see if the effect was specific, right, to a specific internalizing disorder, or general to the whole spectrum, and found evidence for the latter, right? So this looks like that. That's a hazard ratio up there, on that path connecting internalizing to survival. Right, so the idea here is, okay, those disorders, major depression, generalized anxiety, panic, and a sort of neurotic personality style have so much variance in common that they can be thought of as one coherent variable. And those residuals there have no, notable, greater than one or less than one, right? No residual risk effect on survival time, right? The whole effect is being mediated through the general path, right? So that's kind of the point of that paper. Consistent with the idea, right, that these things are part of a consistent, internally coherent spectrum, and that's what's important to understand in terms of understanding, in this case, all-cause mortality. Yeah, so, a little plug for that thing, right, that there's a utility to a transdiagnostic perspective. Maybe you know this already. Mental disorders are not categories in nature. Psychiatrists continue to pretend that that's the case, but there's no evidence to support that kind of perspective. They're conceptualized as categories just owing to tradition. And, the reason that kind of persists is because there's this conceptual hegemony that flows from the Diagnostic and Statistical Manual of Mental Disorders. But the empirical literature illustrates really that these are continuous distributions within spectrums of variation. And you can illustrate that, using MIDUS because there's this evidence for coherence of outcomes. So then a little bit about future prospects before we lead into my compatriots' talks. I think MIDUS is well suited to understanding major secular events. You've already seen another talk today about the Great Recession, right? I think that part is really fascinating, to me at least, right? The ability to really characterize the recession impacts. And in our current data collection, we're studying the impact of COVID on people, right? So that's going to be a unique opportunity within the MIDUS context to understand another unpleasant secular event, right, and its impact on people's health and functioning. And that, I think, emphasizes the importance of these kinds of longitudinal cohort studies. The genomic database is continuously growing. Hopefully I've convinced you that it's not daunting to use MIDUS, right? You can go to the web page, download the data, work with it, enjoy it, publish on it. And we're continually trying to grow, the amount of genomic data that are available. And maybe my last point, right, is that we try to be also part of consortium efforts. So I'm particularly excited because, well, I'm a psychologist. Like, I'm very interested in human personality. And there's a big effort, maybe some of you are involved in this, right, the Reformulated Genomics of Personality Consortium. And that's exciting to me, right, because now we have enough sample size to really think we might be able to unambiguously identify SNPs that are relevant to human personality variation. And the stuff coming out of there is promising looking. I think there's going to be Ted Schwab is the guy who's running this consortium, and I think he's speaking at the behavior genetics meeting this summer with some preliminary stuff that looks pretty compelling. There's a lot of data on neuroticism now, because neuroticism was in UK Biobank, so that helps too. But anyway, like, MIDUS is part of those kinds of discovery efforts also. So that's another way in which we're trying to use the data to hopefully contribute to the social genomic enterprise that is the focus of this meeting. Okay. So I think we can have a few questions before we turn to the next speaker, if you want to do it that way. If there's any questions for me before I sit down. Please.
Do you know there are any MIDUS samples also enrolled in ALPHAs?
Sorry, what was the last? I missed the second part. Maybe the microphone.
The question is about the data. So ALPHAs. So suppose we run a GWAS using ALPHAs samples. We want to calculate PRS in MIDUS, but we do not want to have sample overlap.
Oh, I see. Yeah.
So do you know whether there's samples in the MIDUS also enrolled in ALPHAs?
Right. So MIDUS, I think what you're asking about is MIDUS being used in discovery efforts, right? MIDUS has not been used in many discovery efforts to date. The re-GPC thing is, I think, the first one that I'm involved in. So I'm pretty sure that MIDUS has rarely been used in discovery efforts, which means there's cleanliness there. But most consortia, right, can compute the, PRS's net of a particular sample, like hold it out and then compute the PRS. But yeah, it's not been used in discovery, and so it's clean in that regard, if that's the question, yeah. Other questions? Wendy?
Can you talk about, so there was a Milwaukee subsample to capture more African Americans. Have they, how large is that subsample? Have they been genotyped or any biomarkers from them?
Yes. That's the short answer, right? So the longer answer is, in an effort to diversify the MIDUS sample, there's a Milwaukee oversample, right, where the idea was to go to Milwaukee. That's a city near here, actually. I grew up in Milwaukee, as did you, right? So, and the idea there was to, go to Milwaukee and recruit people who were African American predominantly, right, into an oversample. So yeah, that's another kind of, you can think about it as a subsample of the bigger MIDUS project. The Milwaukee oversample exists.
What wave was that done at?
Well, multiple waves. Like, this gets complicated, yeah. So there are multiple waves of Milwaukee, and yeah, a significant, well, the Milwaukee oversample people who also participate in the biomarker sample would have genomic data. So then what you get into is, everybody in this audience appreciates is, well, what are we going to do about the computation of polygenic risk indices, for example, for this kind of sample where the, LD is different, this whole thing. So we didn't release scores for them, but if you get the raw data, you'll have the scores. So these more sort of contemporary techniques, right, these kind of multi-ancestry GWAS things that are now available, right, to the extent that people are reporting now scoring approaches that are based on multi-ancestry considerations, one could go in, right, and estimate those scores. And also for MIDJA, right, for the Japanese part of the sample, it could be done, in addition to the, African-American part of the sample from Milwaukee. See, there's so much here. It's a treasure trove. Please enjoy MIDUS, and then I'll have done my job well today. Other questions? Please.
Personality data is in it?
Okay, so there's an instrument that measures the big 5 personality traits with some adjectives, right? So openness, extroversion, neuroticism, agreeableness, and conscientiousness are measured with a series of adjectives. And so there's that in pretty much every wave, right? That happened from the very beginning. At around wave 2, I added something called the multidimensional personality questionnaire, which gets at 11 more fine-grained facets of personality. So that I added because the big 5 are kind of like at a broad level of the trait hierarchy, if you will, right? So I wanted some more like facet level stuff, in MIDUS, some more specific things. So yeah, there's also the MPQ. I think that got in there at wave 2. It's probably in there at wave 3. It is in there at wave 3 also, right? So starting with wave 2, you also have these little more fine-grained things. That's very short. Like it's like 3 items per scale. So you could wonder about the reliability, but that's what we did, right? So that has things like aggression, right? Like aggression is a personality tendency that doesn't map neatly onto the big 5 because it's kind of interstitial between neuroticism and disagreeableness, right? So there's stuff like that that's sitting in the MPQ. Yeah. Other questions? Wendy.
So I heard that there's a way you can link external contextual variables to MIDUS participants' data. For example, if I was interested in census level indicators of neighbor disadvantage, how could I get that linked to MIDUS participant data?
You'll need to contact the CIA about this matter. No, in all seriousness, right, you can do it, but you kind of have to do it in like a sealed room, right? I think you can, well, I know for a fact that you can do it because people have. And so you request with the MIDUS administrative core to be able to have access to those kinds of sensitive variables. And I think you probably have to do the analysis on a server that's located at Wisconsin, right, instead of VPNing into it or something like this. I'm pretty sure that there's ways to do it, and that's how you do it, because people have done it. So it can be done. It just would be a special process, understandably. Other questions? Okay. I will turn it over to my colleague, Dr. Marcon.
Yeah, thanks for being here. I'm very happy to talk about my research. So today, let's see if I can do this. So today, I'm going to be talking about work that I've been doing in MIDUS on gene methylation and expression. So I'm going to be talking about the expression data. And in particular, relationships with, this is sort of a mouthful, but psycho-socio-economic variables in aging. The expression analyses I'm going to talk about are actually focused on cognition. I was hoping to talk about some other stuff as well, but that I sort of ran out of, I don't want to say ran out of time, but the servers have not been cooperating the last month or so. So there's lots of facets to aging, and I think this is something that's been on my mind a lot. We talk about aging as kind of a unitary sort of concept in chronological aging or health decline or something like that, but there's lots of different facets to aging. So there's cognitive aging, different physical aging. There's different components to it, and so sometimes when we're talking about we talk about aging as this sort of general process, but I think sometimes it accelerates sort of at different rates for different people for different things. And there's different indicators of aging. So CRP is something that people talk about. Grip strength is another example. These different indicators could be used to indicate, or as indicators of general aging, or they could be used as indicators of specific facets of aging. And if you just think about genomic changes with age, there's a number of things that have been looked at in the literature. So people have talked about methylation markers and epigenetic age acceleration here. People accumulate somatic mutations as they age. This is another thing that we're becoming increasingly interested in. There's also expression changes with age, and so these are all things that change with age that represent different facets of aging and the aging process and things that occur as people get older. So one thing that people have brought up repeatedly, so some of you are very familiar with this, some of you may be less familiar, is this idea of different generations of epigenetic age acceleration markers. And these have been indexed to different things. So some of the earlier ones were designed to predict age. So these are sets of methylation markers that predict chronological age. Some of the later ones were developed by predicting biological or health correlates of aging. And then some of the more recent ones were developed by predicting actual change of declines in functioning or mortality. And these have different patterns of associations that people are sort of still figuring out. So in MIDUS, these are some of the markers that are available in MIDUS, epigenetic markers of aging and MIDUS age acceleration markers. There's actually multiple versions of these available, so some of these have been updated or tweaked in different ways over time. These are the more recent ones. And what I have here are just the correlations between the different markers in MIDUS. So the upper right-hand triangle is not controlling for chronological age. So you can see that these are, in general, much more highly correlated, especially things like Hannum and Phena age and Grimm age. And then in the lower left-hand corner, this is the correlation between these markers controlling for chronological age. And what I put this up here for a number of reasons, but one is to highlight that there's kind of two clusters of epigenetic age markers in this correlation matrix here. So the first ones are this Horvath, Hannum, and Phena age, which are kind of more correlated, and then Grimm age and Dunedin pace, which are also more highly correlated. And those two sets are relatively less correlated with each other. So again, I bring this up in part because it's important to keep in mind that we talk about aging and aging indicators as all sort of being indicators of one thing, but they don't always correlate in the ways that you would think. This pattern is what you see in MIDUS. But when I saw this, I went back and looked at the literature, and you see this in some of the previous data sets that have been published too. Some people pointed this out. The first three indicators are all kind of indexed to chronological age or biological correlates of aging. Grimm age is, the way it was developed is a little bit complicated, but mortality was one of the things it was predicting, and Dunedin pace is predicting decline in health variables over time. So if you think about this in terms of a factor analysis, there's kind of two factors to epigenetic aging in the MIDUS data. One is what I call state, which is sort of age state or health state. And the other one is a factor reflecting epigenetic markers of decline. So you can think of mortality as a very significant and permanent decline, and Dunedin pace is sort of being an indicator of a gradual decline. So in a recent paper that I did with some colleagues, this actually was just in press just the last couple weeks. We did a study where we looked at psychosocial stress and SES as predictors of epigenetic aging, but we wanted to take a more longitudinal approach, and we wanted to sort of treat them as sort of like reflecting a common set of resources and stressors. So a lot of times stress is implicitly embedded in models of SES, so there's this distinction between subjective and objective SES. So you can think of objective markers of SES like household income or assets, but there's also, people also measure like subjective experiences of financial stress or how wealthy you are, independent of objective asset indicators. And I would argue that stress is implicitly like sort of embedded in that sort of conception of subjective SES because it's kind of reflecting someone's perception of SES. And SES is often a major component of psychological stress. So a colleague of mine, Frank Mann, he presented on this in the MIDUS meeting last year. He probably talked about it recently too if he was here. This is a little bit hard to see, but in MIDUS there's measures, so these are all based on rating scale measures of different stressors. And what he showed is that these sort of cluster into these specific forms of stress So discrimination stress is one. It's pretty much what it sounds like, stress due to perceived discrimination. Home and work stress, there's a lot of items on there reflecting like job, work, or family job conflict or home work conflict. Perceived inequality, relationship stress, and financial stress. So financial stress here is a major psychosocial stressor in this model. And they're all correlated, and this is very stable over time. So what we wanted to do is think of SES as sort of part of this general stressor model. And in MIDUS, these variables are assessed repeatedly, longitudinally, so we wanted to look at SES and stressors together longitudinally over the course of the study as predictors of epigenetic aging. So there's 3 assessments here. People were ages on average 49 to 61 across the study. 60% were female, 70% white, and 14.9 years of education on average, or the equivalent. So these were the variables that we were looking at as measures of SES and stress. So educational attainment, so this was the maximum attained education over the course of the study. Mean income, so again, this is mean income over the whole course of the study. Variance in income, so this is how much their income moved up and down over the course of the study, independent of income, so all the analyses I'm talking about controlled for mean income when talking about variance. And then accumulated net assets. And then we had these measures of psychosocial stress. So these were ratings of psychosocial stress, so including general stress, as well as stress, perceived stress in different areas. And again, this was averaged over the course of the 3 time points in the study. So this allows you to get a sense of like actual measured SES and stress over the course of many years, assessed repeatedly. How is it associated with epigenetic aging? So this gives you a sense of what these results look like. And what we found is that, so this is for the decline-related epigenetic factors. So this includes the Grimm age and Dunedin pace markers. If you look at all these epigenetic markers individually, they follow the pattern of the factors. So you can see here that education predicts slower epigenetic aging. Mean income is negatively associated with epigenetic aging. Greater assets is associated with slowed epigenetic aging. And then the stressor variables, so general social stress, inequality, perceived inequality, and financial stress are all positively associated with epigenetic aging. Some of the ones that were less strongly associated were discrimination-related stress, home and work stress, and relationship stress. Those weren't significantly correlated. So here the p-value is the regular p-value, and the q-value is like a multiple testing sort of significance. If you look at the state-related epigenetic markers, though, so the Hannum, Horvath, and pheno-age markers, they don't really relate significantly to any of these variables that when we looked at them. So there's something about these decline-related variables that are more strongly associated with these SES and stress variables. So I also looked at what happens when you control for some of these things. So one question you might ask is, if you have objectively measured mean income and assets and financial stress, what happens when you put these together in a mediation model? And what you find out is that subjective perceived financial stress over this time period relates to epigenetic age acceleration. This is the decline factor, but not actual observed income, mean income or assets, which was kind of interesting to me because I would think it would be the other way around. But it's actually the subjective financial stress that's more strongly related to epigenetic age acceleration. And that could be because the stress is more closely reflecting like actual consequences of financial status or whatever variables are causing the relationship with epigenetic age acceleration. There's also an education, so the EA3 polygenic index is in MIDUS. So I was interested in what happens if you control for that and looking at the relationship between education and epigenetic age acceleration. So EA3 is related to actual obtained education in MIDUS, but it doesn't really affect the association with epigenetic age acceleration at all. So controlling for the EA3 polygenic index really has zero effect on the association with epigenetic aging. So these variables predict accelerated aging in MIDUS. It seems more strongly related to decline-related epigenetic aging markers. And the strongest associations are for things like education, mean income, general stress, and financial and inequality-related stress. So a more recent line of research has been focused on sort of relating these methylation variables and expression variables. So I think someone else was talking about this earlier. The classical model of methylation is that, there's this methylation process that occurs, and it prevents transcription and sort of shuts down gene expression at a site. When you have demethylation, it sort of allows transcription to occur and facilitates gene expression. But there's a lot of studies now showing that this model doesn't quite work the way you think it would, so that there's sites upstream and downstream of a given site that methylation at those sites affect expression. So expression and methylation are not as tightly coupled together as you would think, so they're kind of independent of one another. And I think this has important implications for understanding what's actually happening when we talk about methylation, changes with age and things like that, and more broadly understanding what's happening with genomic changes with age. So in MIDUS, you can look at, there's gene expression, RNA expression, variables in MIDUS. So there's, in the raw data file, there's about 60,000 sites, our expression data for about 60,000 different sites. About half of those have variability though, so there's a lot of sites that don't really show any, there's no variance in gene expression at those sites, depending on the subsample. So the data, what the data actually are, normalized log abundance values. They've been normalized against, so each site is normalized against sort of standard expression markers. So they're really like relative expression abundance. There's also publicly available data, like Bob was pointing out, on these CTRA markers, which are gene expression sites associated with inflammatory and immune response systems. And there's also composites for those available. I'm not going to be talking about those. I'm going to be talking about raw data. So if you look at individual sites, this is kind of the sort of thing that you might see. This is just an example. This is IRF7, which is a site related to signaling in response to viruses and innate immune response. This is kind of a composite of what a lot of the variables we'll see. Like a lot of the sites have abundance values that are kind of look normally distributed. There's also some sites that have these spikes that are at 0 and then long tails. So they're often either very normal looking or very highly positively skewed. And this is kind of a mixture of the two of them, the sorts of things that you see. So sometimes people look at clusters or components of gene expression. So they'll try to identify components of gene expression. Sometimes these reflect sort of functional gene expression networks. There's also been work showing that it's pretty common to find really large components of gene expression affecting just general levels of gene expression or system coherence. And when you look at the MIDUS gene expression data, it's kind of hard to see here, but you kind of see that in this. There's one very large component of gene expression in this data, followed by increasingly smaller, or components contributing increasingly smaller levels of variance to overall gene expression variance in the data. So it's hard to see here because of the scale, but I did some analyses just looking at how well the gene expression data predicts cognition. So in MIDUS, there's a verbal measure of cognition covering a number of different domains in cognition, VTACT. So I looked at this using 74 of these components. 74 was kind of where there seemed to be sort of a natural break in this component variance distribution. So if you take there's a large first component, and that sort of suggests there's this general component of expression. And if you look at this for MIDUS, what you'll see is that if you just, this is pretty simple, but if you just take the average expression level across all like 30,000 sites and just average like how much gene expression is going on for a person and correlate that with age, you find out that there's a pretty substantial correlation with age, relatively speaking, 0.39. It's not super high, but it's not trivial, and some people have seen this too. So there is an overall correlation between overall level of gene expression and age in MIDUS. If you use these components with GLMNet, which is a penalized regression method to predict cognitive functioning in MIDUS, the overall R squared, so this isn't trying to identify sites, it's just kind of looking at the expression, expression correlation, kind of like genetic correlation or the expression correlation with cognitive functioning in MIDUS. An initial training sample, the correlation, the R squared is 0.366. And if you take all of those component weights and the regression weights and just apply them to a new sample, in a cross-validation, you get an R squared of 0.2218, which is pretty substantial in my mind compared to some of the methylation variables that I was analyzing. So I've also looked at comparing the expression and methylation data in predicting cognition. So ASCA is kind of like people who developed GCTA, which is a way of looking at marker level genetic correlations with phenotypes. They have another software, ASCA, which is designed to be used with methylation and expression data. And if you do that, you see that the expression data predicts, so this is using all of the 30,000 raw expression sites, so not the components, but the raw expression sites. The overall correlation with cognition is 0.31, and for the methylation data is 0.21. So I've seen this with some other papers predicting other psychiatric variables where gene expression has the highest prediction of phenotypes, followed by methylation, followed by the genotypes. So I think there's a lot of potential here in sort of looking at relationships between genotype, methylation, and expression and different phenotypes that might be insightful in sort of teasing apart what's going on causally. Some people have used expression data as a way of sort of validating polygenic predictors, which I think is kind of an interesting thing to see if you see the same patterns in expression data that you see with the polygenic analyses. So going forward, I'd like to sort of look at some of this expression stuff with other aging phenotypes, compare methylation and expression, see if patterns of prediction between genotype, methylation, and expression are comparable, if the associations with phenotypes are themselves correlated across these different omics. And then to look at predictors of aging patterns and of expression, sort of to use it as a marker of accelerated aging. And I think that's it. So I'd like to thank Bob Kruger, Steve Cole, and Frank Mann and Colin Freilich for help with everything. So. Any questions?
That last slide about GCTA versus ASCA. The last part, yeah. Can you interpret what you mean by like the GCTA version of expression and methylation data? Like what's the interpretation of two
So you can think of this as kind of like so GCTA you can think of as like a genetic correlation based on marker data.
But so genetic relatedness, is that what you mean?
Genetic correlation with a phenotype.
Yeah.
So like what's the overall R squared in terms of predicting phenotype from genetic markers? But here it's using expression and methylation data instead. So it's kind of technically it's a random effects model where there's sort of when you do this, you estimate like a giant matrix. So it does, as part of the model, it does estimate like overall similarity in expression patterns between each participant.
Can you interpret what similarity in expression patterns between unrelated individuals means? Do you interpret it as similar environmental exposures or something different than that?
I mean, I don't mean to sort of trivialize your question because it's a good question, but causally speaking, I don't know. I mean, it is like similarity in expression patterns genome-wide is what that matrix is reflecting. What causes it, I think it's, you know, it is what it is at some level. I don't mean that dismissively, but, you know, what's causing that is a different and maybe interesting question to think about. My guess is there's a whole bunch of things influencing it. So it could be environmental exposures. You know, one thing like I mentioned, I've been interested in is somatic mutations. So it could be that there's, you know, there's going to be some degree of genetic similarity involved in this as well, probably. That would be going into that. So it's probably a number of different things. I mean, as you go further and further downstream, so to speak, from the genes, there's probably going to be more factors going into those sorts of similarity patterns, I would think.
Thanks.
Okay, thanks.
All right. Hi, everyone. I'm Colin Freilich, graduate student with Bob and Christian at the University of Minnesota. I guess I just wrapped up my 4th year. It's getting close to finishing up in a clinical psychology program. And so this talk is pretty similar to some of the other ones on epigenetic age acceleration and looking at possibly some interesting correlates, possibly some mediation models. I guess one of the things that makes it a little distinct is it's focused on a specific clinical phenomena that I find really interesting, loneliness. And so I'm going to show some work we've done trying to understand individual differences in loneliness in MIDUS, and then move into primarily looking at associations between loneliness and morbidity, and if these epigenetic clocks can help us understand that association. So, loneliness, you know, it's becoming more and more relevant, understood to be more and more relevant from a public health perspective, not just, you know, like target in psychotherapy. It has a wide range of health correlates associated with declining health and across cardiovascular, inflammatory, cognitive, all these different domains, relationships with early mortality. And so, you know, increasingly emphasized from this public health perspective, the U.S. Surgeon General issued this advisory calling it an epidemic. And one, I think, really important thing to note is it's separable from objective social isolation. So it's slightly correlated, I think usually like a 0.1 to 0.2 range, where objective social isolation would be, you know, like how much time are you spending with people? You know, you can, you know, whereas loneliness is an emotional experience, subjective. We can reliably measure it with self-reports. And so, yeah, that'll be the focus here. And yeah, probably not a big surprise to, I guess, folks in this audience, psychologists, that there's a lot of individual differences work using the Big Five to understand loneliness. So this is a 2020 meta-analysis, and I'm pretty sure it's like anywhere from like 60 to 100 studies across traits here, looking at associations between Big Five traits and loneliness. In the bars, it's like the bivariate association in the red, and then controlling for the other 4 traits in this meta-analytic structural equation model in the yellow gold. And what you see is really consistent, relatively moderate link with neuroticism and a negative association with extroversion. So higher degree of neuroticism is pretty consistently linked with loneliness, so high stress reactivity, high propensity towards negative emotion, high level of anxiety, and then a low level of extroversion, also linked with loneliness, so a low degree of positive emotion, low degree of sociability, all these different things we understand to be under that extroversion umbrella. And so those cross-sectional links are pretty well established. One interesting thing I think we were able to do in MIDUS, and it's like my first thing I spent a lot of time working on in grad school, so I guess I like to show it, even if with those error bars you can see it's probably a bit underpowered to detect these differences, but was comparing those different samples Bob was talking a bit about in the prediction of loneliness across the core MIDUS sample, subsetting on just the white Americans in there, and that MIDUS sample, so the Midlife in Japan, and then that Milwaukee African-American subsample. And so, interesting, I think, to look at some of these cross-cultural differences. Hard to conclude too much, but maybe conscientiousness a little more important in the US, and maybe extroversion a little more important in Japan in predicting loneliness, perhaps, and maybe some, interesting cultural explanations you can create based on that pattern of results. So that, I thought, was one interesting thing we were able to do in MIDUS to help better understand individual differences. Also, like Bob mentioned, we have the twin sample, and I think this is, should have wrote it down, but I think it's like 405 or 410 twin pairs, where we were able to look at the covariance between loneliness and the Big Five, and using sort of standard biometric modeling, estimate genetic and unique environmental correlations. So, partitioning out the covariance to understand the degree to which it's associated or due to, underlying genetic influences, or perhaps mediated in the unique environment. I think, yes, right. I think I do have the slide. Yeah, so we did that with the Big Five and that MPQ questionnaire that Bob mentioned, where you can see 11 more specific personality traits, and start to see those genetic and environmental correlations. And I think the interesting thing, or the most interesting thing, to me at least, are those unique environmental correlations across a range of traits, suggesting there is an association between personality, various domains of personality, and loneliness, net of what makes twins similar, their underlying genetic makeup, and their rearing environment. So sort of consistent with a causal claim that these different domains of personality are involved in the development of loneliness over time. And so, I guess building on that one trait I found particularly interesting is aggression, where it really doesn't have a super high degree of genetic overlap, right? Aggression and loneliness you don't think of as too similar, but you do see one of the larger environmental correlations in the group. So, I don't know, you can construct some narrative in your head where they're very different constructs, but perhaps interpersonal aggression, your propensity towards aggression, can lead to the damaging of interpersonal relationships and development of loneliness. Okay, so that was sort of the individual differences work that we've been doing, and shifting a bit to the relevance of the epigenetic markers and trying to understand loneliness health associations. So loneliness and mortality, loneliness and declining health are linked for a variety of reasons. There's evidence of behavioral mediation, so things like low level of physical activity, poor sleep, right? That can help explain some of the loneliness health association. There's evidence of genetic correlation, so some underlying genetic influences. There's not much evidence of reverse causation, but a little bit of work suggesting, perhaps your declining physical health can lead to disruptions in your relationships and the development of loneliness. But you know, the thing I guess we're trying to test or in this project with the epigenetic clocks is the concept of a direct effect, right? Does loneliness cause some physiological signal that you know, leads to the development of chronic conditions and health decline? And so there is, you know, as you've heard today, there's starting to be a lot of evidence linking various psychosocial variables, SES, relationship variables, right, to accelerated epigenetic aging. And I forget who had it in their talk yesterday, but links between loneliness and this conserved transcriptional response to adversity. So pre-existing links between loneliness and inflammatory gene expression, and then starting to be some mediation work that, yeah, everyone's presentations were great, starting to show that, and other work with loneliness specifically looking at epigenetic aging, mediating loneliness's links to cognitive health and cognitive decline. And so that's what we wanted to do here in MIDUS. First, just quantify associations between loneliness and the different markers. And then our outcome variable was like a condition count or like a morbidity variable. So how much are loneliness and these epigenetic markers predicting increases in morbidity. And then we tested a mediation model and a moderation model. So mediation consistent with the impact on morbidity operating through DNA methylation theoretically. And then the moderation model just using an interaction term, sort of consistent with the idea that perhaps your vulnerability to the health impacts of loneliness would be influenced by your epigenetic epigenome. And so, yep, we did this in MIDUS. No surprise there. We were able to use the whole sample of just over 1,300 folks who have who had the methylation profiling, and then this follow-up on, I think this is better expressed on this slide. So we have sort of loneliness preceding the blood draw, the epigenetic measure, and then we have health conditions time point 1 and time point 3. So we do have some time sequencing here where we have loneliness prospectively predicting epigenetic aging and able to use both to predict change in health conditions. And so yeah, all the same clocks Christian showed, clear associations between Dunedin and Grim age, and then Horvath and Hannum, clearly with pheno age a bit intermediary, or moderately associated with all of them. And yeah, I think we can just show some results. So hopefully you can see that a bit. These are just that first aim of predicting the epigenetic markers across the top, and then in the end, it's just like a composite average of the others. With the loneliness row there highlighted. And we ran 2 models, first with just demographic covariates, and then added different health behaviors to see the extent to which those behaviors might attenuate links between loneliness and epigenetic aging. And I put it in some charts that look a lot like the previous charts. But these are just those betas of loneliness predicting each of the different markers. First with the demographic covariates, and then adding in those behavioral covariates. And you can see it's a slight association, but consistently positive. Loneliness predicting accelerated epigenetic aging, more so in the later measures, and slightly, but not fully attenuated by health behaviors. And then moving to the prediction of health conditions. So this here, now the dependent variable is that condition count at the later time points, and we're predicting that with loneliness, with epigenetic aging and with the across the two different covariate sets. And this result, what's highlighted is that mediation path through the epigenetic clock from loneliness through the clock to morbidity. And so there are direct effects of loneliness and some of the epigenetic aging variables, and the indirect effects were really small and non-significant. So not a lot of evidence of mediation, even if it is sort of consistent directionally here as well. Yeah, and similarly, slightly, no surprise there, slightly attenuated by the health behaviors. And then moderation, so same model without the indirect path now, but with the addition of that interaction term to look at perhaps a synergistic effect of loneliness and epigenetic aging on changes in morbidity. And this was, similar. It was significant in Dunedin, which tends to be the one, and Grim age, but Dunedin tends to be the one that picks up, the most signal here. In MIDUS, it seems like. So yeah, perhaps some synergistic effect, perhaps some mediation path, but certainly a little preliminary to make any strong conclusions there. Yeah, so I guess to summarize a bit, loneliness does seem to prospectively predict accelerated epigenetic aging, especially in the second generation clocks, and then attenuated slightly by health behavior. They both were able to predict some decline in morbidity or increase in morbidity, decline in health. And it's a really interesting time point. I should have mentioned this earlier when I was, brushed over the sample. But it's from 51 to 61, so from midlife into, the beginnings of older adulthood, where folks were endorsing the average was like 2.6 conditions on average, 51 up to like 3.5 or so. So we are seeing, I think important discernible increases in morbidity in the sample across this time point. Perhaps some moderation evidence, perhaps I'd say that's a little stronger in this sample than the mediation evidence. I think there was a really good question earlier about, disentangling what these epigenetic signatures are from the outcomes we're using them to predict and understand. And yeah, that's complicated, and I think it'll be really interesting in the coming years when we get more measurements of these epigenetic markers to see how they change, because, theoretically these signals that we're picking up on to predict these outcomes, we don't know when they came along, right? They could have followed the developmental processes associated with the outcomes. And so, yeah, it's certainly too early to say anything causal. But yeah, I think they're interesting for trying to help us understand some of these associations and maybe can tell us a little bit about loneliness health associations. And yeah, I think that is everything. Collaborators, funders, and yeah, happy to take any questions.
Just a quick question on the interpretation of your loneliness to the different acceleration clocks. The estimates, so say 0.1, what does that mean? Is that 0.1 year difference?
Well, it's standardized, so it'd be like a 1 standard deviation increase in the clocks, which, yeah,
But why standard? So why is that the right unit to? show? Like, what does that, what would that mean? 0.1 standard deviation of something?
Yeah.
What is the something?
Right, you could convert it to years. I can't do the math that quickly, but it's not a large degree of years.
And if so, I guess that's how I interpret it, and you have small effect there. I mean, are you also comfortable in saying that the evidence suggests right now that you could rule out loneliness being a major contributor to accelerated aging?
Major, probably, but I mean, I think the effects are similar to what we see across, some of the other psychosocial predictors and SES, right? I don't think any of these things are predicting, huge changes in epigenetic aging, but maybe that's a misread, but I think it's in the range of a lot of these other predictors.
You said that perceived loneliness correlated with like 0.1 or 0.2 with actual objective loneliness. Is that correct?
Sort of. Objective, like social isolation. So like amount of time you spend with folks. Right, there are people who are around people all the time but are lonely. There are people who are totally content to, be socially isolated but aren't lonely. And they correlate but not super strongly.
Okay, and would that mean like hanging out with a group of people that you know, or just like walking through a busy city, and those equally count as objective loneliness? Because it seems, I don't know, it seems really low to me, but I'm just curious.
Yeah,
about
I think
that.
it's lower than a lot of people expect. And yeah, the measures tend to get more at like time you spend with family, time you spend with friends, things like that.
Okay, got it. Thanks.
Yeah.
I forgot my question. So this is the end of 2 days, and so I may get all the studies jumbled together in my head, but I feel like there are several studies, including MIDUS, that maybe has measured methylation at more than one time point, and I feel like MIDUS is one of those. No,
We
just
only
one
have one
time point. Okay, so never mind. Is it a health and retirement study that's done it at more than one time point?
Future Families has
Really?
2 time points. HRS, I'm pretty sure, only has 1.
Okay, because it would be really interesting to me to know how correlated they are across
Yeah.
time, because the critical thing is to measure it at a particular point in time, and then, yeah. So is wave 4 MIDUS, holding the microphone, got them. Is MIDUS wave 4 also going to include more
Yes.
epigenetics? Great.
Someday MIDUS will have all the data. No, in all seriousness, right, like there will be multiple waves. In fact, that's in the aims of the current grant that we wrote, now that I sort of think it through. Right, that was an important aspect of justifying, continuing the study. So yeah, there'll be multiple waves. And I totally agree, it's super important when you're asking, and I think it's very interesting to think about. Right.
You want to see how it changes or doesn't, yeah.
Within persons.
Right, yeah. And then see how that translates into, yeah. Get this away from me.
I would like to comment on Jason's point, the small effect. Like the measurement of loneliness probably just capture transient loneliness, but that may not capture, that captures not only the transient loneliness, but also persisting or prolonged loneliness. So probably the chronic loneliness is very critical for your health, but transient loneliness maybe not. And the questioner asked, how do you feel lonely in the past 30 days? So if you feel only 10 days, if you feel lonely for 10 days, they say yes, but still, it could be a transient, and that may cause or affect your effect size, and that kind of, because the transient loneliness may not affect your health outcome, as well as the epigenetic aging. So if you can distinguish the chronic loneliness from a transient loneliness, I think HRS has a multiple waves asking the loneliness question. So probably it's a good way to distinguish your transient loneliness and the chronic loneliness, have that affect your associated with your epigenetic aging, and this is just a kind.
It's a really good point. In evolutionary theories, loneliness is thought of as adaptive, right? Like a signal to reach out and connect. So transient loneliness isn't necessarily thought to be, toxic for your health or whatnot. Although I don't think there's much evidence suggesting that, and that people who report loneliness in the past 30 days, that tends to correlate pretty strongly with like a broader propensity. Like the UCLA scale is phrased more, never, always, sometimes, right? Like a broader propensity, and they're hard to separate in questionnaires. I think folks are trying to do it in sort of like daily diary type work to get at like day-to-day variations in loneliness, but I don't know that there's a lot of clear evidence that we've separated them well yet.
Thank you. I just have a very general question about these aging clocks, actually. Maybe it's a question also for other folks on the panel. Because we build a lot of genetic prediction models for a lot of phenotypes, but the general sense is many people publish papers and eventually the methods get better and better, and there's a consensus in the field that we should stop using models built from 10, 15 years ago because those are just not as good. But I noticed that every single talk uses many generations of aging clocks as if there's not a sense of which one is better, and they just quantify different things. Which makes me wonder, is it because there's not a sense of how to quantify whether a clock is good or not? And if you have an investment of $5 billion, you can build whatever sophisticated model you want, what would that clock look like?
Well, they're different, and I think we're learning about how they're different, which is why we tend to show all of them. The later ones that are trained on declines in health tend to pick up a lot more signal in various things, demographic factors, psychosocial factors. So they're better in that sense, but you could argue that they're not really quantifying age really at that point because they're predicting health more so than your age. But I don't know. You guys have thoughts on that?
Yeah, I mean, I think that's a good question. I mean, this kind of goes back to what I was talking about earlier, which is part of it is, I don't know how to explain this, but I mean, if I were to ask you what is a good measure of aging, my first question that comes up is, what do you mean by that? Like, what aging in what sense? Like, is it chronological age? Because I don't know that that's exactly what you actually are interested in. And so if you talk about aging with reference to X, Y, or Z, so I think there's a lot of ambiguity about in terms, that's not just related, like if you want to know what a good measure of aging is, you have to have some standard or criteria that you're sort of evaluating that against. And I think if that's fuzzy, it becomes really hard to know what that is. I mean, there's also just been, my sense is, I mean, I haven't been working with these variables that long, but there's just an explosion of literature in this area, and I think teasing it apart just takes a certain amount of time, I guess. And there's also a lot of disagreements in the literature about how to interpret some of these aging measures. So, I mean, just from my personal perspective, it seems like there's just a lot of uncertainty about what a good measure of epigenetic aging should look like, if that makes any sense.
May I just push back a little bit by asking, why do we need this concept of aging? Why can't we just use cognition, blood pressure, glucose, things that we know what they are and what we can quantify? What's the need for something that we call aging without knowing that?
Because they're separate, but I think there's I mean, we're all going to die someday, and there's some declines that lead up to that. So if you look, whatever health-related outcome you look at, there's going to be some terminal decline, and those declines often are systemically correlated with one another. So they're separable, but they're not totally independent of one another. So I think, I think it's a really great question you're asking. I think there should be more research into what do we mean by aging exactly. But I think there is probably some value in knowing, I mean, these things, like I said, these things aren't independent. So if you know one thing about someone's aging status on some variable, it's probably going to be predictive of some other things at some point. I don't know. I mean, these are all things that are related, and just treating them separately, sometimes that's a good thing to do, and sometimes I think it maybe loses sight of the big picture of where people are at in terms of their overall life trajectory. But that's just my perspective on it personally. So.
Thank you.
Can I say something?
Yeah.
I have a little bit maybe to add. I mean, I have so many thoughts about your question. It's just super important. So that your kind of question, right, is what led to a certain extent to the stuff that Christian and I have been doing with just use all the information, right? Because what's going on when people are creating these clocks, right, is they're keying, right, the present methylated at CPG sites to some set of phenotypes, right? And one thing that's remarkable is that stuff does predict age, right? So when you talk about age acceleration, it's like the residual of that, right? So it's how old you say you are and how old your, genome, if you will, epigenome, like implies you are, right? So one thing that's remarkable is that it works in that sense, right? I'm accustomed to nothing works in genetics because the effects are too small, right? So what's been remarkable is that it does work, right? It does predict age.
Can I just switch that into cognition?
Yeah. So that's my point, right? Like that's literally what I think Christian was presenting on. So instead of, because it gets complicated to interpret even, right? So like someone creates a clock, like the Dunedin, pace clock, right? The things that it's keyed to are those things, potential correlates of the clock, right? Because then when you take it to another set of variables and you say, oh, I've discovered something new, because this clock that was keyed to these things also predicts this other thing. Well, this other thing could easily have been in the predictor set that derived the clock, which I think is related to the kind of point that you're making. So you could just take all the information, right, literally, right, in the methylation data and use that information to predict something like cognition, which is what you were presenting on, right, that BTAC measure. So instead of, pre-specifying weights based on something that you keyed the clock to, you just take the information in the methylation arrays and use that to predict the outcome that you're interested in. Now that becomes like biologically less interesting, although that's really not where this literature has been going. You know what I mean? Like there has been less, my impression of the literature, there's been less like these sites predict, these sites don't, and therefore we have some annotation, right? Then there has just been like, look, we can mechanically do it. So I think your line of inquiry is very, very important, right? Because this stuff gets confounded very quickly. Do you see my point? Like if you keyed it to these things, and now you're saying it predicts loneliness or something like that, loneliness could have been in the original predictor set. You could go out right now, take the MIDUS data, come up with some set of things that you think are definitional of aging, and create a new clock. People do this all the time. That's why there's so many clocks. Nobody knows what time it is, but they have plenty of clocks, right? There's just proliferation of clocks in the literature because you can just do that. And so I think that's what's sort of happening in this literature, and the question becomes, well, what does that all mean? Like, what really is the meaning of the methylation data? So I don't know. Those are kind of some thoughts I've had about this.
Thanks very much.
Sure. Look outside. We could go out there. How about that? Well, thanks, everybody.
Let's thank our speakers and go outside.
Yeah.